Good morning, everyone. I'm Dr. Burns, the director of the Kawasaki Disease Research Center, and we're going to start on time, even though people are slowly trickling in. We have a full program this morning, and to be fair to the parents who were here on time, and to all of the people who hopefully are joining us on the internet through the amazing efforts of our research assistant, Matthew Vehar, and our other research assistant and Kawasaki disease brother, Peter Lillian. We're going to be streaming this live on the internet and hope that all the technical issues work out. So it's my pleasure to welcome you today to our 5th Kawasaki Disease Parent Symposium. This is something that we do every year, and it's gratifying to me as the director of the center that we actually have new things to tell you every year, which means that we're making progress. So if you picked up one of the sheets that shows the order of the talks that we'll be doing today, we're going to have talks in the morning, then there's going to be a time for questions and answers, and we're going to have volunteers passing out index cards and pencils. And what we'll ask you to do is write down your questions for us and then pass those in, and we'll go through those at the end of the morning session, the first morning session, and then at the end of the second morning session. There'll be a break in the middle for refreshments, for meeting each other. I hope you will reach out to each other and share some stories about your experience with this disease. So I want to start by acknowledging all of the hard work by the people that you see up here on the screen. These are the fabulous members of our Kawasaki disease research community, and they've all made amazing contributions and worked very hard so that we can make progress in this disease. Our spiritual leader is Dr. Kawasaki, of course, in Japan. I was just recently in India where they had a web stream from Tokyo of Dr. Kawasaki greeting everyone at the Kawasaki Disease Conference in India. That was amazing. Next year, we'll try to get that set up here. But he's doing well at the age of 88 in Tokyo. And here's a picture of Dr. Tremblay and Joan Pankari, who runs our Kawasaki clinic with Dr. Kawasaki in Tokyo on a recent visit. So the world that we live in can be thought of as being divided into two halves. One encompasses the clinical research that we do. That means research directly with patients. That means new treatments, and Dr. Tremblay is going to talk to you about that today. We have the other half of our world is what we call bench research, which is with test tubes and tissue culture and working with DNA and RNA in the laboratory, where we're trying to generate better understanding about the disease that then is going to inform the other half of our world. Because of course the goal of research is to make this world a better place for all patients with Kawasaki disease through a more profound understanding of what the disease is and how we can best control the inflammation and keep the heart safe. I wanted to share with you some of the most recent information from Japan, where they have the largest number of Kawasaki disease patients in terms of an attack rate. So the birth rate in Japan is falling, and yet they had almost 14,000 new cases last year. And this translates into a statistic that 1 out of every 85 children in Japan will get this disease. So certainly not a rare condition in Japan. In terms of the United States, we have absolutely no accurate data about how many patients there are. This is not a legally reportable disease. As I think the current Ebola crisis has pointed out, our public health infrastructure is really wanting. It has been underfunded for many years, and Kawasaki disease is one of the things for which there just isn't any money allocated to actually make an effort to know how many patients we have and how big the problem is. However, here in San Diego County, we've done a really good job of counting all of the patients. And in fact, through collaborations with Balboa and Kaiser, the only two other medical facilities that care for inpatients with Kawasaki disease, we've been able to ascertain that we have about 100 new cases every year here in San Diego. And if you take the most recent projections for our children under 5 years of age and do some back of the envelope calculations, that means that 1 in every 2,000 children here in San Diego will get Kawasaki disease during their childhood. These are the actual numbers from Rady Children's Hospital, where we take care of all of our patients. And you can see that we take care of between 80 to 90 new patients every year. And I think there's a slight increasing trend. We don't know whether this is really that there are more children being affected or just that the recognition is better. So Dr. Tremblay and I spend a lot of time reaching out to pediatricians in the community and trying to educate people about how to find Kawasaki disease. But you'll be hearing some very exciting new information from Dr. Tremblay about diagnostic tests, because I think what we ultimately need to do is give physicians who may not be pediatric specialists better tools so they can find these patients. And I think we're making significant progress in that regard. I think most of you know that there's a genetic predisposition to Kawasaki disease, and the attack rate varies by ethnic group and racial groups. So we see the highest attack rate among our Asian descent population, and we have a very large both Vietnamese and Filipino community here in San Diego, and so we see many children of those different descents. The African American community is also disproportionately affected by Kawasaki disease. However, we have a small African American community here, and to study this problem better, we are now partnering with Emory University in Atlanta, Georgia, where there's a large African American population, so that we can better understand the genetics in that population. If you haven't signed up for our genetic study, we need your help. We can't do this without you. There's a sign-up sheet outside. I hope during the break, if you're not already participating in our genetic study, you'll take the time to give us your email address so that Deanna Shearer in our laboratory and Erica Berry will be able to contact you and send you a little kit. It's done by mouthwash, no blood drawing necessary. And we would be very grateful for your participation. So I also think that most of you in the audience know that without treatment, 1 in 4 children are going to experience really devastating changes to the heart. Now, we can keep all of our patients safe, and we're going to be hearing a lot more about that after the break from our adult Kawasaki disease team. That's Dr. Lori Daniels and Dr. John Gordon. But these are children, if they've had heart damage, who are going to need to be under some kind of care from a cardiologist for the rest of their life. And obviously, if we could prevent that, we would like to do that. In terms of the breakdown of the population that we have here at Rady Children's Hospital, we're currently running, even in the best of all possible worlds, with what we consider to be the perfect first-line treatment, about 7% of our patients are going to have aneurysms. And in many cases, that's because when they come into the hospital, they already have early damage to the coronary arteries. There's another 20%, or 1 in 5, that have transient dilation, meaning when we do the transthoracic echo, the sound wave test across the chest where we measure the coronary arteries, we can see that they're bigger than they should be, but then they come right back down to normal. And Dr. Daniels is going to talk to you a little bit about what we think some of the implications of that may be. So 70% of the children have quote-unquote normal echocardiograms, and we'll be addressing again later today about whether or not there's recommended follow-up or what we think about the outcome in adulthood for people who have been quote-unquote always normal. So this is a study called CT, standing for computed tomography, a CT angiogram where dye is injected through an IV. And then we have a fancy way of imaging the heart and then reconstructing the image in the computer. And the yellow arrows point out the very abnormal part of the vessel, the coronary artery aneurysms, that are a big risk for clot formation, which would cause a heart attack, or over time for narrowing, which can also result in a heart attack. So obviously, both problems that can be managed, but need very careful follow-up. So we get a lot of questions from our local community of parents, but also from around the country, and in fact from around the world, about how children should be followed for Kawasaki disease. And the real answer to that is we haven't had enough experience over time with this disease to have really robust answers to that question. But everybody is taking care of these patients, and we have to agree upon some follow-up that we think is reasonable. And of course, these recommendations may change. So I just want you to remember that we're giving you recommendations on the best information that we have now, but because we're continuing to study this disease, continuing to do long-term follow-up, what we're telling you today may change, and so we invite you to come back every year to hear the latest in terms of information about what we think the appropriate follow-up should be. I sit on the National Committee for the American Heart Association that is in the process of coming up with new revised guidelines. The last guidelines from the AHA were published in 2004, so it's been 10 years now. And we are trying very hard to finish this up, and so I hope that next year we'll have a new version of the guidelines. What everybody can agree upon is that you should diagnose and treat this disease as quickly as possible, and the standard treatment for uncomplicated Kawasaki disease is a large dose of intravenous immunoglobulin plus aspirin. All children should have an echocardiogram at baseline while they're in the hospital. They should not be allowed to leave the hospital until that happens. And discharge is considered to be safe when the fever has resolved for 24 hours. I think those basic principles pretty much everybody can agree on, and there's nothing controversial there. We also have good data to support, and this is in a collaboration between our center here and Boston Children's Hospital. We're among the two largest centers in the United States, and combining forces, we've put together data that suggests that the first clinic follow-up should happen within 10 to 21 days after treatment with IVIG, and that should consist of a clinic visit with some blood testing to check on the level of inflammation and how it's resolving, and an echocardiogram, the sound wave test to measure the arteries. At that point, our data suggests that if the echo is normal and if the inflammation has resolved, there really is no need to do another echo 1 month later. That was something that we did in the past, but the analysis most recently of our data from the two centers suggests that that's probably not necessary, that if everything's normal on that baseline echo and on the first clinic visit, that we don't need to do that second visit during the subacute period. However, we and the American Heart Association are recommending coming back at 1 year for a repeat echocardiogram. And this is sort of a safety net to make sure that we haven't missed anything, to check in with the family, see how things are going. If the echo is again normal, so now we've had 3 normal echoes, then we are currently at our center following patients every 5 years. We get distraught emails from around the country from parents saying, that's not how my child's being followed. They've discharged me from care. They don't want to see my child anymore. And we have to say that insurance is now backing off and not paying for these extra visits. And there is no data to support that we need necessarily to see your child back at these 5-year intervals. For us, we think that this is a safety net, that this is a significant disease. This is about your child's heart, and it's about their heart for the rest of their life. And we invite you to come back every 5 years for a visit. However, we are getting pushback, as I said, from the insurance companies, and the AHA guidelines that will be coming out will not recommend further follow-up for the population in general. Again, this is only for children who are normal at all 3 looks. But again, we're doing things differently at the research center here. We want to see your children back every 5 years, and we hope that you will come back. If the echo is abnormal at any point, the follow-up is tailored to the individual patient, and there are no specific rules about what the appropriate follow-up should be. It's different for every child in that situation. And finally, we come to what we call the exit visit. So at age between 16 and 18 years, we want to see all of your children back for the battery of tests that are listed here. And you're going to be hearing this afternoon from Dr. Gordon about the CT calcium score and what that involves and why we recommend it and why we think this is fabulous reassurance for $100 that you have to pay out of pocket right now because insurance companies are not paying for it. We think it's a fabulous way of buying some reassurance. And if all of these tests are normal, we are now discharging these young adults, these 16 to 18-year-olds, discharging them from further care. Again, whether or not that's going to turn out to be our same recommendation next year, you're going to have to come back and find out. But right now, that's the state of the art. So I'm going to turn the podium over now to Dr. Tremblay, who is going to share with us some exciting new information. And again, write down your questions. I don't know if we've passed out does everybody have an index card? Yes? Okay, so write that down, and then we'll be collecting those, just passing them down to the end of the row at the end of the series of talks before the break.
And thank you for joining us today. Can everybody in the back hear me okay? Good, okay. I just want to say hello to everyone that's joining us by live web stream today, as well as hopefully some colleagues in Latin America. We have a large Latin American network that extends all the way starting from Mexico to Argentina and Brazil into the Caribbean, and I've invited them all to join us today. So if they're out there, bienvenidos. So good morning, everyone, and I've been given the task to talk about creating a diagnostic test for KD. And even though we think of Kawasaki disease as one disease, we know that it varies, as Dr. Burns mentioned, in terms of the level of cardiovascular disease that can affect children. However, if there's one theme that I think we hear with all of our children, it's the fact that their parents really wish that there were a diagnostic test out there, because most parents feel as if they had to take their child to multiple providers before somebody made the right diagnosis. And maybe even some of you in this audience, certainly we know of many kids that are missed and come to us after their children have peeled, or worse yet, have cardiovascular disease that goes undiagnosed until they have a cardiovascular event like a heart attack. So given the importance of this, I'm going to show you the many, many ways that we've been trying to address this issue. All right, so we certainly know that early treatment reduces the risk of coronary artery abnormalities. Here is the picture with that white arrow of an aneurysm, which we would love, of course, to be able to avoid. And later this morning, I'll be talking to you about treatments, particularly for children with aneurysms. But we still know that it's being missed. And so currently, we really feel like we're only diagnosing the tip of the iceberg. In the US, there's about 5,000 to 6,000 cases that we know of with Kawasaki disease. But we know that it's probably much more than that, unfortunately. So which child has KD? Here are a number of pictures of children that have symptoms that could look like Kawasaki disease. And in fact, it's really only this child here that actually we diagnosed with KD. So the current dilemma for the providers that we have in our community, whether it be a pediatrician, family practitioner, an NP that may see you, an ED doc, urgent care, is that they're seeing lots and lots and lots of kids, but it may be only one out of very many that actually have Kawasaki disease. And so that's a very difficult task to take on. In our own emergency department, I wanted to put up some statistics. You know, San Diego has about 3 million people. We have over 90,000 visits that come to our emergency room annually. Of those, about 7% of them are being seen for fever. And there's a population of those that are certainly going to have Kawasaki disease. So our job is to try to help physicians figure out which one in that group has KD. And in fact, we developed a slide because if I take a diagnostic test to a company and I say, I want to develop a diagnostic test for KD, they're going to say, well, who's going to be using this diagnostic test? What's the incentive for my company to develop a test for 100 kids in San Diego annually? Well, the reason that they want to develop a test is because it's going to be used in a much larger population, right? The population of kids that are going to be tested is much larger than those that actually have KD. So our job is to develop a diagnostic test that has a very good way of finding kids with KD, and it's going to be used for a large group of kids, but finding really what we call that needle in the haystack. So I was thinking about how to show you all these data, because we have a lot of different ways that we're trying to figure out, and basically all roads we want to lead to Rome. But sometimes I feel like our providers feel like Rome is in a million directions. Like, you know, where is Kawasaki disease? within our patients. So the approach we've taken is this. I'm going to be talking to you about 4 different approaches that we have right now for developing a diagnostic test so that we can have all of our roads leading to this diagnosis. And we've been able to look at this from many, many avenues and with many collaborators around the world. So it's a very exciting time for us. I think last year I stood up here and I talked about just that blue box with the biomarkers, but you can see that we've made some significant progress also with our cell phone app, which I'll also be talking about. So let's look at the biomarkers. So biomarkers are proteins that we have in our bodies that allow us to hopefully identify a particular disease, okay? And so this is really a very hot topic in medicine. And in fact, what we're trying to do is to be able to apply this particularly to Kawasaki disease. And our ultimate goal is to develop a diagnostic test that distinguishes the child with a fever and another disease from a kid that has fever for Kawasaki disease. So this started many years ago. And when I look back at the slide, I said, wow, it was really back in 2010. And in fact, this, our whole work with biomarkers started back in 2010 with one of our moms who put together a KD Fund Day to raise money in order to develop a diagnostic test. And maybe some of you were even with us at that time. And here's a list of the original 89 analytes that we looked at, which is really a laundry list of inflammatory markers. Okay, so these are biomarkers of inflammation. And through that KD Fund Day, by having our families stomp the ground and run around to a lot of local companies, they raised $25,000 from us. And that was the beginning of the development of a diagnostic test within our group. And we're still incredibly grateful for the families believing in us. And frankly, a lot of the money that's come to our research center to develop the diagnostic test has been from many in this audience and from the outreach that many of you have done to raise funding for us. So we're incredibly grateful to you for this funding that we've developed. So we took the $25,000, we did some testing initially to look at these 89 analytes, and what we discovered is that if we're just looking at inflammatory biomarkers, we really cannot do a very good job of distinguishing between kids that have a fever because of KD and kids that have a fever for another reason. So what we found was that we were really hoping we would find within these 89 markers something common that could be easily measured, but that was not as easy. That task would have been wonderful, but it wasn't as easy. Instead, we've had to move on, and things have gotten a lot more complex. So let me talk to you about some other work that we've been doing. This is work that we've been doing with a company called Momenta that's based in Boston. And we're moving on to less common markers, okay? And what you see here on your left-hand side is what's labeled as febrile controls, and on the right are children with acute Kawasaki disease. In fact, many of these kids may be your own that are within this mix of patients. So again, another reason and another way that you guys play into our research is just that initial blood draw that we always say, do you mind if we take a little bit of extra blood? Well, that goes into being able to develop these diagnostic tests. So what you see here around the perimeter of the circle are different markers that are much more novel, I guess is the word, or less common than the inflammatory original list of 89 that we had, okay? But what's really telling is when you overlap these maps here. And now in the red, we're looking at Kawasaki disease. Brown is our febrile controls. And in looking at it this way, you realize that peaks start to come up in the KD group. And so a lot of the work that we're now working on with Momenta is to learn more about these peaks, learn more about these particular biomarkers that may be distinguishing our Kawasaki disease patients from febrile controls that don't have KD. And if you read some of the orange writing, you'll see that some of these are actually what we call acute phase response. So again, inflammatory markers like that list of 89. But there may be some things within the coagulation cascade. And we all know that our kids go on aspirin to prevent coagulation from happening. That may be critical in distinguishing kids that have fever and that are febrile controls from our KD patients. So we've been making some definite waves in the biomarker world, and we continue to pursue this. But that's not enough, okay? So the second approach to a diagnostic test has been clinical and laboratory data. Now, I alluded to this to you last year, but let me just go a little bit more in depth about this test that we've developed. So what we have here is a collaboration that we're incredibly proud of with Stanford University. We've been able to pair with their bioinformatics and their statisticians up there. And this is really creating what Dr. Burns and I call a KD expert in an app. So when I see a patient with Kawasaki disease, I'm immediately thinking about what's that child's age, what's their sex, what's their ethnicity, what symptoms or signs of KD do they have, what does their blood work show us, what does their urine show us. All those things go into my mind, and then I think of what's the probability that this child has Kawasaki disease. Do they need to come into the hospital for an echo? Do they require immediate treatment with IVIG before their echo's even done? And so those are all the things that Dr. Burns and I think of when we see a patient in the ED, when we get a call from a pediatrician, when you call us from your own home saying, I'm worried about my kid having KD again. That's the process we're going through. So if we do that, can a computer do that? And so we have now created an app that we have with Stanford that someday I'm hoping really looks like this little heart there on everybody's iPhone or your Samsung. And here you have a listing of all the different things that we collect, the demographic data, the clinical data, the laboratory data. And that gets put in and it gives you a probability of how likely this child has Kawasaki disease. Now, how have we done this? Again, with your children's data. So we have taken our patients, your children, their data, in addition to the children that come in through emergency room that we enroll as febrile controls, and we've made the computer figure out who has KD and who doesn't. All right, and we've done this with about 95% accuracy, looking at data that we have currently in our computers. But now the task is to figure out how well it works prospectively. So here is how it worked for us. And you can see that we did pretty good initially, but we had to make it better. So this is a paper that we published in 2013 in Journal of Pediatrics. And it shows on your left-hand side, you have FC, which is febrile control in the green. Red is Kawasaki disease. And you can see that the peaks are separated, but there's some overlap. And that overlap in the middle are the kids that the computer can't figure out if they have KD or not. All right? So you see here on your right-hand side, the febrile controls and KD, and you can see that we did pretty well, but it wasn't great. It was able to pick up nearly 2/3 of the febrile controls and nearly 3/4 of the KD patients. But we need to make this better and get to what we have right now. And I'm going to show you something very exciting that's happened in the last year. All right, this looks complicated, but I'm going to break this down for you because I think it's really cool what our collaborators up at Stanford have been able to do. So there you have on your left-hand side, number 1, all right? And these are 2 groups, 2 cohorts with a lot of children, 533 KD patients, 318 febrile controls. That's one group, and then the other group has 268 Kawasaki patients, 161 febrile controls. So that's a lot of kids. These are all data points that we've collected over the years from our patients and through our emergency room. And what you see over here on your right-hand side is what we call the first clinical model. And that's what I just showed you in the previous slide. That's the one that we published in JPEDS, which was good, but it wasn't good enough. But what we've done in the last year is the second clinical model. And what that does is it takes what you see on your left-hand side there, that group 1 through group 4, and these are looking at breaking down the kids into different groups. So do they have 1 clinical sign for KD? Do they have 2? Do they have 3? Or do they have 4 and 5 clinical signs? And what I mean by that is, do they have just a rash? Do they have rash and red eyes? And all the combinations that can be created by the computer. And so what our collaborators at Stanford asked was, okay, we take all this clinical data, we take the laboratory data, but we can only predict maybe 3/4 of the kids that have KD. What if we take the kids in the middle, this yellow group, the undecided, and we apply a second model where we now look at that group in the middle and we say, can we develop a model depending on how many signs of KD they have, and can we do an even better job at figuring out who has Kawasaki disease? And by doing these 2 models in tandem, we do much better. And in fact, we did so well that we're able to pick up a lot of our kids, nearly everybody that has Kawasaki disease. So out of that over, I think it was 801 children, 769 of them we figured out that they had KD, all right? Now, there are kids that we missed, and that's always going to happen. But the more important question to me as a clinician with that child is, did we miss the kids with heart disease, right? Because this is not looking at an echocardiogram. You have to remember, and as most of you know, when your kid comes into the emergency room, there's no echo technologist running around ready to do your echo. That gets done the next day. So I want to catch kids without knowing their echo data. And so how did this test or these series of algorithms work? So it turns out that the kids I really care about are the aneurysm kids. And of these over 800 children, 31 of them had aneurysms. And our algorithms so far caught every single one of those 31 kids that had aneurysms. It also did really well with our dilated kids. It only missed a handful. So this is very encouraging. Okay, we have over 95% sensitivity in picking up kids with KD. And that's fantastic. But now we got to see if it works in the real world. Okay? Because we've done this all on retrospective data that we have collected in our computers. And now our task is to see how this works in emergency rooms in the county. So what we've been able to do, and in fact, a very, very recent NIH grant that I just sent in 3 days ago, is we're hunting for funding and looking for funding to do this outside of our computers and to do this real live in real world. And we have 3 emergency rooms that have bravely signed up, and I'm going to be stomping the pavement to get more involved. But we have Pomerado Palomar Emergency Room, Scripps Institute in North County, as well as Rady Children's, our very own hospital that's going to be doing this in their emergency rooms and using the algorithm real time to see how this works in real world. So we hope that it continues to do well and that we're able to eventually have a clinical algorithm on an app on everyone's iPhone. But we know that even with the best laid plans, that may not work beautifully. So we need to continue looking for more. And that's exactly what we're doing. All right. So we're looking at, this is changing paces for you guys here. This is definitely different. The electric nose. Anyone heard of the electric nose before? All right, I hadn't heard about this till our Stanford collaborators told us about it. It's not this. Okay. But what it is a technology that's out there to make in a computer what your nose does beautifully well. Okay? Taking a smell, having it processed in your brain, and you figuring out that you're smelling some Brazilian coffee. It's taking all these signals and trying to figure out if you're given a whole different host of coffees, which one's the Brazilian coffee, to have the computer smell. And it turns out that this technology is being used in many ways, and that our collaborators at Stanford had heard about this. One of our, Bruce Ling up there, had been traveling back to his home country in China and heard about the electric nose technology and thought, well, you know, I am into developing diagnostic tests. And so he actually started using it for urinary tract infections to see if he could develop the technology for figuring out which kind of infection the child had to be able to focus on the right antibiotic treatment from the get-go in a kid that presents to the emergency room. And then he thought, well, heck, I'm collaborating with UCSD Kawasaki disease folks. Can we use it to look at urine in Kawasaki disease patients? So in fact, that's what we're working on right now, is to see if we can develop a technology that diagnoses KD based on urine. So how has he been doing that? So I'm going to walk you through this. And I think it's important just to understand that even though what I'm talking about seems very distinct and independent, we really feel that at some point, all these points are going to converge and kind of lead to Rome, right? So our job is to figure out which of these tests work, in what order we should be applying things. And this takes into account a little bit about this philosophy. Here you have all the way on your left here, a patient with Kawasaki disease. All of our kids get urine, and eventually our goal is to be able to have a point of care test that we can develop. The computer modeling, or the computers here that you see here, are really information that maybe it'll involve our clinical app. that is also part of this technology as well. And you can see that it's really fascinating that the goal is to be able to have high throughput printing of a chip that you can put some urine on, you can upload into a computer, and maybe in combination with our clinical algorithm, it tells you the probability of a child having Kawasaki disease. So this is what we've been, our latest and greatest, and we're very proud of a lot of this work that's going on. And this gives you an example of some data that we have so far. Each column here is a patient, and each row is an individual marker. So our collaborators up at Stanford took a whole long list of markers and looked at them to try to figure out. It turns out that they were able to distill it down to 11. And you can definitely just see here, just by these dots, that the pattern is different within a lot of our Kawasaki patients than the febrile controls. And eventually our hope is, just as I pointed out in the previous slide, that there'll be something maybe like a urine dipstick that can be used in order to be able to put some information together and figure out the probability of KD. But you can already see that we've come quite a long way here. It doesn't capture everybody. There are patients certainly there that have black in their entire column, so they're not being captured. And so it may be that we do need to combine some of our technologies to put everything together. However, this is all great if the provider is thinking about Kawasaki disease. I don't need to ask our parents in the audience that saw a lot of physicians, how many of you were with your child in front of a provider that should have diagnosed KD that didn't? So our diagnostic tests are good if the provider thinks about KD. It's like the rapid strep test, okay, or the mono test, which is like the only two that are really out there so far for pediatrics. They're good if your provider thinks about it. Now, the strep test, frankly, gets used now, like even if you come in with a broken toenail, all right? You may come in complaining of one thing and your kid still gets a rapid strep. Well, maybe that'll happen someday with our Kawasaki screening. Frankly, I would not be upset if that happens because that means that our clinicians are thinking about it. But this is a proverb that the eye does not see what the mind does not know. So if your provider is not thinking about Kawasaki disease, it doesn't matter if I'm able to combine our app with our algorithms, with our biomarkers, with our urine tests, they'll never run it. So what are we going to do about that? This is where natural language processing comes in, all right? So we are very fortunate here at UC San Diego that we have one of the lead groups that works in bioinformatics in the entire country, led by Lucila Ono Machado, who we've been working with her for the last few years, where Kawasaki disease is actually one of her pilot studies on a big grant. One of the people in her group works on natural language processing, known as NLP, there in the middle. And this is a way for us to help the computers think of language as humans do, to ask the electronic medical record to find patients that need to be thought of as possibly having Kawasaki disease. Can we help the computer detect KD? All right, so let me walk you through this. So what we've done in collaboration with this group here at UCSD is we are trying to teach computers to think like clinicians. So here's a history of a child, a 2-year-old male seen in the ED yesterday, vomiting for the past 5 days, presenting for fever, vomiting, decreased oral intake. Parents say that the rash started 2 days ago, just on cheeks and on the back of the neck. So what we're trying to tell the computer to do is to pick out words and phrases that look like Kawasaki disease, all right? So it's picking out fever. How many days ago? 5 days. It's picking out polymorphous exanthem, which is a fancy way of looking, or it points out rash, which we also call polymorphous exanthem. It's pulling out things, for example, like eyes became bloodshot yesterday. So that's the conjunctival injection that we see with KD, and the lips are red, cracked, and dry. That's the oral cavity changes that we see with KD. And so what the computer programmers are doing is they've taken notes from our emergency room from children that have fever for other reasons, not KD, and KD patients. And they're trying to teach the computer to go through and read the clinical notes so that it can figure out which kid may have KD. And by doing so, our hope is that this will be able to actually alert physicians that this child should be screened for Kawasaki disease. All right, so remember, these children may just be coming to a clinic. They may not even have had labs drawn yet. All right, so, but our hope is that we will then be able to have the computer look for kids that need to be screened. So the process here is tagging the clinical signs and symptoms in the note, giving it a score, and then alerting the clinician and the clinical provider to suspect KD. And so eventually, we have a lot of electronic medical records in the city of San Diego. Our hope is that we can have a little ping come up that says, did you think about Kawasaki disease? And that may alert the physician to actually do some labs. And with that in mind, then maybe they can do the diagnostic algorithm after that and put everything in. All right, so just in the last couple slides, I just want to be able to thank all of you. So our parents have really come to the rescue for this. I have to say that since the very beginning, we have certainly been applying for extramural funding. I'm hoping the NIH will bite on this last application, but the NIH has actually to this day never funded this work. We have had funding gratefully from private foundations like the Hartwell Foundation, and I've been funded also by Robert Wood Johnson Foundation, but we're most grateful to our families for raising nearly $250,000 through CrowdRise last year. And a lot of that money is what is supporting a lot of this work currently. I just want to end by thanking everybody that's been very much involved with this work. I just encourage you all to continue telling everybody that you know about Kawasaki disease. The more that you put it on your Facebook, get it out there in social media, the more children that we will be able to identify. My task is to be able to have a series of tests that will be out there to really help our clinicians so that we don't miss any children with Kawasaki disease. And thank you very much.
So people at the back of the room, come on down. There are plenty of seats here. Dr. Tremblay gave us a fabulous overview of the research ongoing here about how we're going to find the Kawasaki patients. But another topic that's near and dear to our heart is what causes Kawasaki disease. Obviously, if we figured out the cause of Kawasaki disease, that might lead us not only to a very specific test for the disease, but potentially, and this is a long-term goal, to its prevention through vaccination or other means. So I'm going to share with you some of the crazy work that we've been doing that has led us to suspect that Kawasaki disease might be transmitted on the wind. So you have seen perhaps this concept before that the world is divided into genetically susceptible and genetically resistant children. We've talked already about the genetic predisposition. And when these genetically susceptible children come in contact with agent X, whatever that turns out to be, or maybe X1, 2, and 3, maybe there are multiple causes, but in any case, these children and only these children respond in a way through their immune systems to create the disease that we recognize as Kawasaki disease. And we suspect that the rest of us are also exposed, but we just don't react to whatever this is in the environment. So people have looked at a number of different microbes, from fungus to bacteria to viruses. And so far, it's been very disappointing that we haven't found an agent that we can blame for triggering this disease. So it occurred to those of us who take care of the patients that this was a very seasonal disease. And because it was seasonal, we thought we should join forces with climate scientists to try to answer the question why it was seasonal. And so we teamed up with the Japanese epidemiologic team at Jichi University in Japan, and then Dan Kahan at Scripps Institute of Oceanography. And again, this is a little bit of the serendipity of research. This is how things happen, that you happen to be at a university that's next to one of the greatest oceanography institutes in the country, and we took advantage of that. And the work that Dan did was to take the Japanese time series, which is very complete and very robust, and then determine the average incidence in terms of number of cases per day for each month in each prefecture in Japan. And the results of that research were this compelling series of maps where you see January designated as month number 1 and December down in the other corner designated as month number 12. And where you see red, that means that that specific location in Japan was experiencing higher than the average number of cases for that particular location. And where you see blue, and if you focus on the months now of November and October as well, you see it's very blue. And that means that there's a lull in the number of cases. Kawasaki disease never completely disappears, but we clearly recognize these seasons. And so you can see that a peak season is in the middle of winter, January and February. Then it drops off, or the signature becomes a little less distinct. And then you end up in the middle of the summer with another peak, and then this real lull in the action in the fall. So very seasonal disease. Why is this? We then went on to look at clustering of cases, and we showed that that was true both here in California as well as in Japan. And we wanted to go on to explore whether there were climate variables that might explain this kind of seasonality. And we performed research that was looking at time series in Japan, in Hawaii, and San Diego. And this required the collaboration and was really spearheaded by a group in Barcelona. So again, to give you the sense that this activity of research is a global effort. This isn't something that we do in our little silo here at UCSD, but it involves collaboration, in this particular case, with the entire planet. And so the group in Barcelona was very active in the past in studying climate disease relationships, and they agreed to jump in and tackle Kawasaki disease. So their hypothesis that they were testing, the idea that they wanted to test, was that a regional scale climate pattern might precede the onset of a KD cluster. And it's hard to transmit or convince you that this was an aha moment for this climate scientist. I mean, he literally jumped off his chair when he saw this association. He had been sifting through lots of data, looking at precipitation and dew point and temperature and all the kinds of variables that you might think of with climate. And when he looked at the directionality of the wind that was associated with these large epidemics here, it leapt off the computer screen at him that there was a very, very strong association. And what the association was that the direction of the wind, when there were high numbers of cases shown in red here in Japan, the direction of the wind was from China. And when the epidemic subsided, the wind came from the Pacific and blew in this direction across Japan. So this is the country of Japan here. And the wind changed directions when the number of cases fell. And this was true for all three of the major epidemics in Japan, and in fact was true for the seasonal variation that they saw each year. So we're talking about winds that are about 5,000 to 10,000 feet above, or meters above the surface of the earth, so up towards the jet stream where airplanes travel, jet airplanes travel. So not the wind that you necessarily feel on your face here. And when they did this modeling with a time series in the bottom from Hawaii, the middle is San Diego, and the upper one is Japan, the red shows actual number of Kawasaki disease cases, and the blue line is a mathematical derived vector of the wind and the directionality of the wind. And you can see that the red lines and the blue lines are overlapping, thus suggesting that there's an important association between these two events. So we had an historic meeting in Barcelona with the team there and representatives from both Japan and the United States. And we came up with the hypothesis that these winds traveling at this height above the surface of the land carry an agent that when it's inhaled by our genetically susceptible children causes KD, and that this KD agent was being carried on dust particles originating from some area in Asia. So the big excitement, and some of you may have seen this treated in the lay press, it got quite a bit of coverage. And in fact, if you go online and just do wind and disease, you'll get many, many references to this work. This was published in June of this year in the Proceedings of the National Academy of Science, which is a very prestigious journal, and it is the summary of the work done in collaboration with the Barcelona team. And I'll try to explain to you what exactly they did. I'm not a climate scientist, I'm a medical doctor, but this has required learning a whole new set of language to be able to communicate with my colleagues. And I have to say it's been incredibly satisfying and enjoyable from a professional point of view to just delve into a whole new area of science. So what they did, if you can imagine, they took wind and treated it as a box, okay? They took a volume of wind, and using satellite data, they said, okay, this is a box of wind overlying Japan at a time when there are lots of cases. Where was this wind 3 hours before? And where was this box of wind 3 hours before that? And they did what's called backtracing of the wind until they found the place on the planet where the wind again slowed down. And when wind slows down, so wind never stops, you may not have stopped to think about that, but wind is always moving. But it speeds up and it slows down. And when it slows down, there is exchange of what the wind is carrying. That rains down, those little dust particles. And the things that are on the surface of the earth can also loft up into the wind. So there's an exchange process. And so they wanted to know, where's the next place that this wind that we think contains the Kawasaki agent, where did it slow down? Where did it pick up its payload? And when they did the backtracing of the wind, modeling the 3 big epidemics in Japan, this kind of analysis that was done for 257 individual days, every 3 hours, going backwards in time. It took about 12 months of supercomputing time with parallel processors. This is a very computationally intensive kind of work. And what they saw was this brown region covering the area where the wind slowed down. And as you can see, here's Japan here, here's Korea, and here's China here. And these dark brown spots in here designate very, very intensive crop locations, agricultural crops. And what they did was to actually target that the origin of the particles that were being carried to Japan came from these 2 provinces in China. Now, this is obviously a hypothesis. This is something that will have to be tested. And these 2 regions in China are not necessarily the source region for the world. They're probably independent source regions all over planet Earth. But this is a very good laboratory, a very good situation for us to study specifically the wind coming from this region and going over Japan. And as it turns out, this is the wind that may also have affected your children and caused their disease. So we think of dust as little round dots or particles, but when you look at a dust particle of the size that you could actually breathe in all the way deep into your lungs, this is actually what the dust particle looks like under the electron microscope. So you can see there are plenty of nooks and crannies in there where various molecules or even organisms could hide and safely be transported across very large distances. And an example of such a long-range transport of an infectious agent is from the Saharan dust storms that originate, as you can see in this little video, over the northern part of Africa. And that wind travels carrying these dust particles across the Atlantic Ocean. And when it hits the Caribbean with the warmer temperatures, the air slows down and it drops Aspergillus sedowii, which is a fungus that is not pathogenic. It doesn't cause disease in humans, but it makes the fan coral in the Caribbean very sick. And those of you who are avid snorkelers or divers may have noted or be aware that the fan coral is dying out in the Caribbean, and it's being killed by the spores of this fungus that are coming from Africa. So here's an example, not in human medicine, but in the biologic world, of a long-range transport of something that was pathogenic. So we collaborated with Hiroshi Tanimoto at the National Institute of Environmental Sciences in Japan. They have research aircraft there. They do a lot of monitoring. They're very worried about the air that's coming over to them from China because of all the pollution. So they have a whole system of aircraft that does this kind of monitoring. We were able to get some time on the aircraft, and actually the pilot was paid by a grant from Barcelona, but we're really needing more funding. We've applied to the National Institutes of Health for more funding. We'll see where that goes. But this aircraft was taken up and it flew in the direction that's specified by that red line there, and it was flying exactly into the direction of the wind from China, collecting the dust particles onto filters that were handled very carefully so as not to contaminate them from things on the ground that we weren't interested in. And those filters were put on dry ice and flown to the laboratory of another collaborator, Dr. Ian Lipkin at Columbia University, who is an internationally renowned specialist in finding new agents. And he has a special laboratory there at Columbia. It's called a BSL-3 containment. And what that means is it's a very high safety level. They can work with Ebola virus there and other things. We weren't worried about being anything on the filters being dangerous to us. We simply wanted to work in an ultra clean environment so we didn't contaminate the filters. When we isolated the nucleic acid to look for living things, we wanted to be very careful. So what was blowing in the wind? Well, we did an analysis of looking at the nucleic acid in there, and we found lots of different species of bacteria, lots of different species of fungus in there. But the thing that really leapt out at us was that we found yeast. So this is Candida. It's a kind of yeast. It's not the yeast you make bread with, but it's a first cousin of that yeast. And it was very surprising to find this yeast because although these kinds of experiments have been done for other reasons in other parts of the world, no one has ever found yeast aloft in the tropospheric wind. So this was the first time, and if you type yeast and wind on your Google search, you will get tons of articles about this work. So we're not saying that yeast caused Kawasaki disease, we're not saying that this is what your child inhaled, but we're saying this is the beginning of walking down this path to try to understand what's there, and this is a clue, and this clue needs to be pursued. And the way we're pursuing it now, these airplane flights are incredibly expensive for the pilot, for the plane, etc. So we have a land-based system now, which is sitting on top of a roof at the Kanazawa University Climate Center, which is located here on Japan. So we're right across from the source region. Here's the putative source region right up in here. And if we blow up this little region here, this is the Noto Peninsula that sticks out into the Sea of Japan. And the air sampler is here. The filters are being changed once a week and being carefully collected in a sterile way and frozen for later analysis. And we've applied, as I said, to the NIH to try to get some funds to analyze it. We can't do any of this work without funding, and that's where we're stuck right now. So all the filters are being collected, they're being saved, but we need the money to go forward and do the analysis. So how does this affect what happened to your children here in San Diego? This is what we call Kawasaki wallpaper. And what Dan Cahan and his team down at SIO, Scripps Institute of Oceanography, did was to make this giant wallpaper that goes way back in time. But here you're just looking at the year 2011 and 2012. You're looking at each month of the year. And across, you're looking at every day in that month. So there are 31 spaces across. And where you see a yellow 1, that's where there was one Kawasaki patient who had the onset of their fever on that day. And where you see 2, there were 2 children who had their onset on that day, etc. And so what you can see is that there are these dry spells where we had no Kawasaki disease patients. And here's a big long one. And then there were what the climate scientists call swarms. I guess that's a climate term. It's not what I would have chosen, but swarms of KD patients who come in within a short period of time. And so they did an analysis of what is the circulation, the ventilation coming in from Asia, from this region in China, because it comes across the Pacific Ocean towards us. Is there any relationship between this pattern on the wallpaper and what the winds are doing? And shown in this last slide is a series of maps that are actually a composite of a large number of these dry spells, is what you're looking at here, that were overlaid from analyses going all the way back to 1995 to the present. And let me walk you through what you're looking at here. So first of all, the red star here is San Diego. And we start at -4 days before the beginning of one of those dry spells that I just showed you. And this is -2 days, 0 to 2 days into the dry spell, okay, and 2 to 4 days kind of in the middle of the dry spell. And what you're looking at here, here's Japan, here's China here. And this big orange-red blob in the middle is a high pressure zone. So you're looking at pressures in hectopascals, and the details in terms of the actual climate science don't matter. But what you're looking at is a big high pressure zone that is blocking all of the circulation coming in from China. So when we have, and these little black dots here are a statistical analysis saying that this is an anomaly. So it turns out that before every dry spell that we have, there is an anomalous, meaning unusual, not usually observed, an anomalous circulation pattern where we have absolute blocking of all the wind coming in from China. When we don't get the wind from that region in China, we have no Kawasaki disease here in San Diego. And we're now extending this analysis. We've been working with sites all along the West Coast, and we hope to go all the way up to the Canadian border and model the circulation. It gets very difficult to model circulation of the wind over landmass, but it's relatively easy to use satellite data to model the circulation patterns over the ocean. And so that's what we're in the process of doing. So I hope that I can stand up here next year and give you yet more exciting data. But I think we're on a path. We don't know if it's the right path. That's why we call this research. It means you search and then you research. And so we're in the process of doing that. But I think this is a very promising avenue. Certainly in the 30 years that I've been working on this disease, I'm more excited about this than other clues or hints that we've had in the past. So let's see where this goes, and we'll give you an update next year. Thank you. So it's my pleasure now to introduce KD Olson. I'm sorry.
The parents of Raphael Canlis.
Raphael Canlis's parents, are you here? Yeah?
Yeah, Raphael.
He's asking for mom or dad.
Very nice to meet you. I'm sorry you didn't come up.
So you have your
Microphone on, you're all set?
I believe so.
Okay. So Katie's going to tell us
us about an exciting opportunity to try to solve the problem of we don't know how many Kawasaki disease patients there are, and we don't know how to find them to participate in our research studies. So thank you so much for coming.
Great, thank you. Well, my name is KD Olson, and I'm a research manager with Sanford Research. And we are the host of the Coordination of Rare Diseases at Sanford, shortened to CORDS, and that's what I'm going to be speaking about today. Just like to thank you so much for having me today. I'm excited to be here and tell you about the CORDS registry. I'm excited to meet my partners that I've only spoken to through email and on the phone. And I have to be honest, on an entirely selfish note, I'm excited to be here because when I left South Dakota yesterday, it was about 30 degrees. So it's wonderful weather here. I'll give you a quick overview of what we'll be talking about today. I'll give you an introduction to CORDS, our mission and goals, how our partnership with KDF is working, how we enroll patients and families, how we collect data and manage it, and then the future steps for anyone in this room if you'd like to enroll today. So a little bit about Sanford Health. Sanford Health is a healthcare organization located in the Midwest, as you can see. We're one of the top 50 largest not-for-profit healthcare organizations in the United States, and we are the largest rural healthcare organization in the United States. CORDS and Sanford Research, which is where I work, is located in Sioux Falls, South Dakota. We were established at the Children's Health Research Center by Dr. David Pierce, and it is a central international patient registry for all rare diseases in all ages. So a question I get asked a lot is, what is CORDS? Why do you have CORDS? What's the point of this whole registry? Well, CORDS developed out of the mission to accelerate research into all rare diseases. And we tried to figure out how to do that, and we decided to create a patient registry, which creates a database of patient contact information and clinical data that we can provide to researchers to study rare diseases. We also provide a mechanism through which researchers can contact patients who are interested in being part of research studies and clinical trials. A little bit about the CORDS team. There's only 3 of us on the slide up here, so we look small, but we are actually a much bigger group when you consider our infrastructure. There's Dr. David Pierce, our principal investigator, myself, and Austin Letcher, our senior research associate. But we're supported by a scientific advisory board, our IRB, our research IT team, our general IT team, our marketing team, and the Sanford Health Foundation, which provides quite a bit of our funding for the CORDS registry. I just want to touch on the scientific advisory board I mentioned. This is a group of wonderful individuals, all on volunteer basis, but they provide guidance for CORDS. They direct us toward researchers and potential partners. So I mentioned the internal team at Sanford Research, but probably the 3 most important stakeholders I want to mention for the CORDS registry. First of all, being patients and families. The CORDS registry wouldn't exist without patients of rare disease. And what we want to provide to those people are a database for learning about research studies and clinical trials for which they might be eligible. The second stakeholder are patient advocacy groups, where we partner with them and we can work to develop a disease-specific registry and collect data that's specific to that rare disease. And then our last stakeholder are researchers who can access the database to look into rare disease and all rare diseases across the board. So what is a rare disease? A rare disease is a disease that affects less than 200,000 people in the United States, which doesn't actually sound like a very large number, but when you take it into effect, you'll see there are over 7,000 rare diseases. So approximately 30 million people in the United States and 350 million people worldwide are affected by rare disease. People in the room understand that because you yourselves have been affected. About 80% of rare diseases are genetic in origin, and over half affect children. So why do we study rare disease? We study rare disease because it's very hard for patients and families to connect with researchers in their home states because there are so few patients. Researchers can't study 1 or 2 cases. They need to study larger groups of people. So that's what CORDS does. CORDS provides a database of hundreds, thousands of patients for researchers to study. And then our ultimate hope is that by studying one rare disease and many rare diseases, we'll find common therapies and treatments that can be applied across the board. What is a patient registry? What is the CORDS registry? The CORDS registry is essentially a research study. It's a program to collect, store, receive, retrieve, and disseminate information on patients with rare disease. So why do we need those? We need those so that patients and families have a place to come and get information, a place for researchers to find information about patients and their disease, a place for drug companies looking to develop treatments, a place for patient advocacy groups to come to develop disease-specific questionnaires. So why is CORDS unique? CORDS is unique because it's essentially a first of its kind. Many patient advocacy groups have their own unique registries, which is great. We don't fault those at all. But CORDS is unique because we accept applications from anyone with any rare disease. So essentially, we don't just accept KD patients. We accept KD patients, patients with CDLS, Klippel-Feil syndrome, anything. Anyone can apply, or enroll, I should say. We also collaborate with patient groups, as I said, to create disease-specific questionnaires, which gather specific data on your specific disease. How are patients and families involved? Patients and families that decide to enroll today will contribute information by filling out a questionnaire, and then you can receive information about your group, your rare disease, and any opportunities for research studies that you might be eligible. So how do researchers access CORDS? Well, they submit a brief application to our scientific advisory board, which I pointed out earlier. They can log in and access the de-identified information and then conduct any sort of analysis they want and identify patients that they want to contact. At that time, they work with the CORDS team to contact participants. CORDS personnel does the contacting. Researchers never get your information. And then at that time, you can choose whether or not you want to be part of the study. How is KDF involved? KDF recently partnered with CORDS to create a customized questionnaire on KD. So it has KD-specific questions on it in addition to the normal CORDS questionnaire. It was IRB approved, and then CORDS at Sanford Research has the software infrastructure in place to house that data. This is just a quick slide on our other partners to show you that we do have many partners. KDF is just one of our 40 partners that we work with currently. And then I want to talk about our enrollment options really quickly. First of all, there's online enrollment. There's web-based enrollment platform where you go online to our website, fill out a quick screening form, literally won't take you more than 2 or 3 minutes. You'll be assigned a unique username and password, and then you'll go online to fill out your questionnaire. If that's not your interest, we can also mail you enrollment forms, or we can do in-person enrollment today. I have about 10 forms that I could work with you if you're interested in doing that. So online enrollment, what that looks like, if you look at the screen right here, that's our screening form. Participants go online and they fill out this quick screening form. Once we receive that, we'll assign you a username and password. You'll receive a link from us to click. You'll go in and fill out your questionnaire. You'll see an informed consent, which basically tells you the details I've just told you today. And then what you can expect is an annual contact from us requesting that you go in and update your information. That's a quick screenshot. That's our informed consent at the top, and then that shows you what the link to the questionnaire looks like. So the CORDS questionnaire, I promise you, doesn't look too scientific. It looks a lot like any other questionnaire you filled out before. The first part of it's going to ask a lot of your contact information, how you want your information identified in the future, whether or not you'd be interested in being contacted by researchers, family history, and then the symptoms and diagnosis that you've received. This is my opportunity to brag for just a moment. The CORDS registry is doing really well, actually. As of November 1st, we do have over 1,600 enrolled active participants, and we're representing about 300 rare diseases. This is just a little graph that shows you CORDS was founded in 2010. Our enrollment was fairly slow until about 2012, and that's when we first launched online enrollment into our registry, which then you can see it kind of jumped from there. This is a very intense slide, but basically the only thing I would ask for you to take from it is that our registry is very secure, your data is safe. We follow all the same standards and requirements that Sanford Medical Center, one of our hospitals, follows for your electronic medical records. Your data will not be lost, it won't be accessed by anyone who shouldn't be getting at it, and you don't need to have any worries about that. And then one last point I'll make is that coming soon, within the next year, we will have a new registry software. We're very excited. If you think back to a couple of slides, I told you there's a 2-step enrollment process. So you'll fill out a screening form, we'll assign you a username and password, and then you'll go in and fill out your questionnaire. Within the next year, that will all be rolled into one step. So you'll fill out a screening form, and then you'll be taken directly to the questionnaire. So that's something to look forward to. Does anyone have questions?
So Katie, I think what we'll do is we'll take questions during the break.
Sir.
And thank you so much for that presentation. what we'd like everybody to do now, we've got some runners at the back here who are going to go down the aisles and collect questionnaires. Foot, if you could come, and Deanna, we need Deanna Chisato to collect questions. If you pass them to the aisle, we'll pick those up. I can actually come and collect them. Not sure where everybody else is. Got any questions for us? So good to see you. Questions? Oh, thank you. Okay. Okay, Joe, with that. Any questions? No?
Let me take this moment just to mention something that came to our attention recently for an opportunity for parents. I'm sure you guys all want to go to Hawaii. So every 3 years, we are very lucky to be able to have an international Kawasaki disease meeting that we hold that has investigators from around the globe that work on Kawasaki disease. It's our intense think tank. We're there. Even though we're going to be in a beautiful place, I'm sure we're going to be in a cold hotel room for 3 days to interact and exchange information about the latest and greatest that we've learned. It's our chance to be able to share what we're talking about today with collaborators from around the globe, find new collaborations, work internationally. There is a parent component to that meeting. You're all certainly invited to join us. I know the KD Foundation just sent this to us. The conference dates are on there, February 3rd through 6th. It's going to be at the Hilton Hawaiian Village in Honolulu. And I think we're going to be, hopefully this has already made it out as an email blast, but otherwise we can send it out. I just wanted to put this up to let everybody know about the meeting. And also, I believe that the registration fee is going to be very nominal for families. And there will be a parent component to this where the KD Foundation is going to be able to have parent meetings while they're there, and you're also certainly all invited to go to all of the scientific meetings and to meet our own friends and colleagues and collaborators that work on KD from around the globe. So we're very, very excited. This happens only every 3 years. Dr. Kawasaki always is able to if his health as well, is always able to join us. So it's an invitation for you to join us as well. And there's more information, as always, about KD on the KD Foundation website. So that's your place to go for this. All right?
Okay, so here are some for you to
you.
start with. And I'm going to go answer a page.
So why is there such a large increase in the Hispanic population in 2010, 2013? And it's a great question. So it turns out that the data that Dr. Burns put up shows that we have a number of children that are Hispanic. Probably about, I think, a third of our patients are Hispanic. So it's great that Dr. Burns, I'm actually a native Spanish speaker. Dr. Burns speaks beautiful Spanish as well. So that's one of our assets that we can provide to our families. There's been a little bit of increase. I think that maybe it's part surveillance and awareness and bringing that out. And that's one of the reasons why in this last year we set up our Latin American network. And although I won't be talking about that today, I'm happy to talk about that further with any of you individually at the break times, because we do have a huge collaboration throughout Latin America. But there are definitely a lot of kids that are of Hispanic origin with KD. I'm not sure that the numbers have gone up all that much recently in particular. Oh, so someone asked for more clarification about febrile controls. And we call what I put up on the slides is FC, febrile controls. So we have this great system that we have through emergency room, whereas if you bring your child to our emergency room and your child has fever and one other symptom of KD and has had fever for at least 3 days, we are constantly on the hunt for blood samples and also urine from children that ultimately are not diagnosed with Kawasaki disease, right? Because I cannot create a diagnostic test in a vacuum for Kawasaki disease. I have to compare it to something. And the comparison is not a healthy child on the street. The comparison is the child in the emergency room who does not have Kawasaki disease, who looks like they may have KD. So that's a very important thing. And it's a detail, but frankly, it's why so many diagnostic tests for KD and a lot of work for KD has not worked, is because people don't have the right comparison group. Our comparison group has to be children who do not have KD that may look like they have KD. Frequency of CT angiograms, it's very patient-specific. What I'll just say is how often do we need a CT angio? Dr. Burns and I will do CT angiograms on children with aneurysms when we think that the CT angiogram results
Hi, how are you?
Oh, can someone tell her that she has the microphone on? When we think that a CT angio is going to change the care of our child. Now, we have made huge strides with our CT angios in our hospital, and actually people fly in from all over the country for this, where we give very low-level radiation. Okay, everybody else gives a lot of radiation for CT angios. We give very, very low-level radiation, which is, you guys should all feel comfortable if your kid needs a CT angio at Rady Children's. But we only put the children through the anesthesia and the risks of it if we think it's going to change their medical care. So that's very individualized, and we try to keep that to a minimum. Can echo detect blood clots? It can, it's not perfect for very large aneurysms. In fact, and some of our families in the audience know this unfortunately, for very large aneurysms, we can be fooled into thinking there's a clot there, when in fact it's just that we couldn't get enough dye into the large aneurysm to be able to tell whether there's a clot or not. So echo is not the best modality, but sometimes we can see clots by echo. Any long-term behavior problems in KD kids via Facebook? This is fantastic. I'm so happy people are sending in stuff. What? Oh, okay. An adult with KD analysis said there's no behavior problems. All right, so what I will say is that as what we know, what we believe from our kids, and this is 1,500 kids that we follow, the two of us follow in our clinic, is that we don't think there's any long-term neurological or neurobehavioral effects from KD. Many of you will know that the trauma of being in the hospital, the inflammation from having Kawasaki disease, makes your child not seem like your child for anywhere between 2 and 6 weeks initially. Right? That is quite common in hospitalized children. It's aggravated by the inflammation of Kawasaki disease. But we really don't think there's any neurobehavioral long-term consequences. Now, things like the CORDS registry, which will follow kids over time, things like Dr. Lori Daniels' adult KD study, where we're looking at long-term and comparing people with KD to people that don't have KD, may elucidate other information, but we really don't think that of all the things our KD kids that get, neurobehavioral is not one of them. If a KD child gets KD a second time, is the case milder? Wow, okay, so it's interesting. So I'm not sure that I can speak to that, and I'd have to, it's a great question, but I don't think I have all the data at my fingertips to be able to say whether or not we think it's a milder case. I'll tell you that my experience has been with the few that I've had it more than once, that they haven't had bad heart disease the second time as they may have had the first time. But it's an excellent question, which I think if Shazada wants to write that question down, I think this is a student project for a summer. See, you're helping our research. Skin problems, yes. In fact, the skin problems are such in KD that we collaborate with a pediatric dermatologist. So we won't be talking about it today, but atopic dermatitis, eczema, psoriasis are the two things we do see. We see some very severe psoriasis in very strange patterns, sometimes more on the face. It doesn't seem to be permanent, but we do see it. And if your kid has had KD recently and is having skin issues, please let us know. Dr. Winnis Tom is our go-to dermatologist at Rady Children's that sees and takes care of these issues. Does KD or KD treatment have any impact on a child's immune system? In fact, we think that IVIG, because you're getting so many great antibodies from the thousands of donors that have created IVIG, we think that it's actually an immune boost for a moment, for some time. IVIG lasts about 11 months in the body. We don't think there's any long-lasting effects about immune issues in our children, although it's rare, but we do have some kids that we share with some of our immunologists as well as our rheumatologists that happen to get another disease like rheumatoid arthritis or some of the periodic fever syndromes, but that is not common. IVIG in particular does not cause any significant lifelong alterations to the immune system. want to make sure that I'm leaving time for Jane to also have questions. I had KD as a child. Should I be concerned about my children? So KD is a genetically inherited disease. The children of a parent who had KD are at a higher risk of having Kawasaki disease. But what I always say is even though they have a higher risk, they also have a great advantage, which is that the parent knows what Kawasaki disease is. And so that we have had families that have called us and said, I know my kid has KD and I had KD, or we've had families come to us. And so this is like, you now have a KD doctor in your household. And you will not miss Kawasaki disease in your own children or in your grandchildren or in any of your Facebook friends when you see those pictures posted on Facebook and you diagnose somebody else's kid. Or I just had one the other day where the grandmother of a KD patient was in the supermarket, and there was a mom there with her kid that was sick, and the kid had red eyes, and I don't know if the kid ever ended up having KD, but the grandmother stopped the mom and said, your kid could have Kawasaki disease. So you're always teaching, so thank you. So, but for that child, if their parent has had KD, they are at higher risk, but they have the advantage of having a KD expert in their household. Do you get many KD patients presenting with atypical symptoms? So incomplete KD, we certainly do see that. Do you feel many are missed due to this? Yes, unfortunately. And one of our goals that we have to eventually test with all these diagnostic tests is we need to make sure that our diagnostic tests are able to capture not only the complete KD, because that hopefully isn't being missed, although it sometimes is, but our goal is to be able to create diagnostic tests that even catch the incomplete kids, because those are the kids that are missed. So I'm going to now open it up to Dr. Burns, and I'm certainly going to be at all of the breaks as well, so feel free to come over and ask questions.
There's some more questions here. So I'm going to take about 5 minutes now to answer questions, and then we're going to have our break. And of course, people can come up to either Dr. Tremblay or myself and ask your questions individually if we don't get to them. We are going to have another question and answer period at the end of the presentations by our adult cardiologists. And so that'll be another opportunity to answer some of your questions. And some of these actually presage that presentation. So there's a question about longitudinal studies on KD patients and how have KD patients done as adults. So there are some adults here who had Kawasaki disease in childhood, and we are the only group that I'm aware of in the United States, actually in the world, that has had an organized study beginning in 2009 of adults following Kawasaki disease. And you're going to hear the results of some of those studies from Dr. Daniels and Dr. Gordon in the next session. So I'm going to let them take over, but the adult KD study is ongoing. At the present time, we've lost our funding. We were funded by the Macklin Foundation for 4 years. We've now lost our funding. And so we're only able to continue on as a registry. So we're doing a health questionnaire, and the purpose of the health questionnaire is to try to answer exactly this question. If we're not tracking these patients and not following you on into adulthood and further, then we're never going to have the answers to these questions. So it's a modest beginning. We've studied about 160 young adults so far. We're hoping to really increase those numbers. We want you all to have your families, your children stay in touch with us as they age up. And eventually we're going to have answers to the questions of how these people fare in adulthood, but there'll be some information about that in the next sessions. Then there's a question about the change in tissue of the coronary arteries, and does it have scarring over time? Right now in children who have had all normal echoes, there's no evidence that there are clinical problems related to Kawasaki disease later in life. But the big caveat there is we studied 160 patients. Actually, about a quarter of those have important heart changes and damage. And everyone who has had heart damage from Kawasaki disease is guaranteed to have some kind of issues in adulthood. and must be followed, must be managed. With management, they can be safe, they can do well, but they must be followed up. So in the past, we had problems with children being released from care because their aneurysms had remodeled. And while that makes the blood flow better, it is never again a normal artery, and those children are also at risk for problems in adulthood that again can be managed and everybody can be safe, but they have to be in continuous care and surveillance. Another question about chronic health problems, not related to KD, but other chronic health problems. We do think that children with KD probably have some differences in their immune system. Whether or not that predisposes to autoimmune or other diseases later in childhood is exactly the reason that we're doing this study, that we're doing the questionnaire, and we really encourage all of you to participate. There's a sign-up sheet out there. Matthew Vehar, who's been doing our audiovisual, is our research coordinator for that project, and both Dr. Daniels and Dr. Gordon are going to talk to you about that, but we just really need your participation. We can't do it without that. The question about Kawasaki disease and eosinophilic esophagitis. This is a disorder that is becoming increasingly diagnosed and recognized, kind of like Kawasaki disease. It's a chronic disorder. And strangely, we have an overlap where we have several patients who have had Kawasaki disease and then develop eosinophilic esophagitis. We don't know if there's an overlap. However, Seema Sevis, who's a researcher and physician here at UCSD, is the person who takes care of the eosinophilic esophagitis patients, and we're partnering to try to understand if there could be some link between those diseases in some individuals. So here's some questions about the WIND project. So in regards to the WIND affect your KD analysis of patients in the US. So the question is this WIND that we've been talking about coming from China, is this really what's causing Kawasaki disease in the United States or in Europe or anywhere else? And the answer to that surely is no. If this WIND theory is right at all, which remember, this is a hypothesis that needs more testing. But if it's correct, then there are independent source regions around planet Earth. And these source regions may be regions of intense agricultural practice. Remember that Kawasaki disease, as far as we understand right now, is a new disease after World War II. After World War II, there were a lot of changes in agricultural process and procedures. There were the introduction of herbicides and chemical fertilizers and differences in the types of crops that were grown. This region in China that we've been studying had massive intensification of agriculture with cereal crops. There's the burning of biomass from these crops. So there are all kinds of things that changed after World War II, changed in many places around planet Earth, and may be related to the cause of Kawasaki disease. So we think there are multiple source regions. China is only the source region perhaps for Korea, Japan, Hawaii, and the western coast of the US. So do we think that an increase in air pollution in China could be part of the problem? There is not a big signature in the filter that we analyzed of specific pollutants, but again, this is very early on in the research, and we're hoping that the land-based filters will give us a lot more material to work with, and we'll be able to specifically address that. I think the thing we're most interested in is heavy metals, particularly zinc or mercury or some of the other heavy metals that can become aerosolized and are used in agricultural chemicals. So we'll definitely be looking at that. But it's, the terrible brown air that you see in Beijing, that's probably, it's not those particulates that are probably linked to KD. And there's a question about how many white children get Kawasaki disease every year. So we have children of European descent and we have children of Latino descent. And because of our population base here in San Diego, of the 80 to 90 patients that we take care of each year, the majority, meaning more than half of those, will be children of Latino descent. The next largest group will be children of European descent. We try to get away from the term Caucasian, since that really should just refer to people from the Caucasus Mountains in Russia, and that's really not what we're talking about. So as we get more sophisticated with genetics, we try to get a little more precise. We take care of a large number. The smallest number of patients that we will care for in terms of absolute numbers are children of Asian descent. However, as a representation of the population, the attack rate is much, much higher, because only 12% of our population here in San Diego is of Asian descent. So children of Asian descent are way overrepresented among our Kawasaki disease population, and we are quite sure that that's because of a specific genetic predisposition among that population. So someone wants to know, how much funding do we need for the WIN study? So please come up and talk to me at the break. I'd love to deal with you more specifically on that topic. But really, any funding that you give is appreciated and used. We do have a UC Foundation account, and at that account, the University of California takes a 6% haircut off your donation when it goes in for administrative purposes. The rest of it comes directly to our laboratory for discretionary use. It is not used for funding my salary or Dr. Tremolet's salary, but it can be used for funding other people in the laboratory. In terms of the WIN study, what we really need is increments of about $100,000 to buy the reagents and fund the people to actually do the nucleic acid analysis. So this is expensive business. This is not research comes with a big price tag, but any incremental donations are appreciated and welcome. And you can do that at our website. You can do it through the KD Foundation and earmark it for us. So a number of different ways online that you can donate.
All right, thank you everybody for your attention. And hopefully you had a little bit of time to mingle at the break. Everybody should have new postcards at their site so that they can write down more questions. I'll be talking about treatments, new treatments for KD, and then we're going to have 2 talks by our adult cardiologists. So I actually need a pointer. Here we go. All right, so we're all familiar with the standard treatments for Kawasaki disease with IVIG and aspirin. But I want to talk to you about newer therapies that we're doing. And in order to do so, I just want to set the context quickly for a clinical trial. So a clinical trial will usually compare the old versus the new to see what new treatments, and if the new treatment is better or the same or worse than the old therapy. We do what's called randomization when we do a clinical trial in many ways, or many times, because randomization is like the flip of a coin. I, as the investigator, can't decide whether your child's going to get the active treatment or not for a study, because I'm probably going to be biased in some way or other. So there has to be a level of randomization in clinical trials as well. And finally, there's statistical analysis that then, independent of my own observations and my own biases, is the way that we determine whether a new treatment is better, worse, or the same as a previous treatment. So with that in mind, I want to talk to you a little bit about one of our initial trials that we were very happy to publish this last year. And this is about infliximab. So we got to thinking about the inflammation that happens in Kawasaki disease. And it turns out that there's a protein called tumor necrosis factor alpha, also known as TNF-alpha, which is a molecule that's made by the body that promotes inflammation. And I'm going to represent it in the next few slides with this pink oval. And TNF-alpha is like throwing gasoline on the fire in Kawasaki disease, because we know that it's elevated in children with KD. So there's a drug out there called infliximab, or Remicade, which is used in rheumatoid arthritis, which decreases inflammation by inhibiting TNF-alpha. So again, here you have these pink ovals, and then you have these little Y-shaped antibodies floating around. That's the infliximab therapy that attaches to the TNF-alpha and takes it out of circulation. This is a therapy that's been out there and exists. I saw a postcard from someone here whose child was in our original 2006 infliximab trial. That's the initial one that was done to show that it was safe. And then after that, what actually brought me on board to to our KD Research Center was in designing a trial to see if infliximab would be the right therapy to be giving children with acute Kawasaki disease. So we designed a trial which was infliximab plus IVIG for the initial treatment of KD patients. This was sponsored by the Food and Drug Administration. We were the first ever awarded FDA orphan disease grant that was awarded to UCSD, and we're very proud of that. And we are going to be applying for another grant just like this in February for one of the other trials that I'll be talking about in a moment. This is a 2-center study that took 4 years, nearly 200 kids. It was with us in Columbus, Ohio with Nationwide Children's Hospital. And the goal of the study was to decrease the rate of resistance to initial treatment of KD and hopefully prevent aneurysms in children. And here is the product of all that work. We were very happy to be able to publish in Lancet, which is one of the premier journals in the world for biomedical research. And here's our paper that we published just this year on this particular study. I'm going to go through the details of it. So the first thing is that whenever you do a clinical trial, you have to prove, or you have to choose before you ever start the study, what you want to demonstrate. So you have to kind of make this decision before. It's not fair to make it after you get all the results back. You have to choose something at the beginning. So what we chose is that we were looking for the difference in treatment resistance rates between children that just got IVIG and the children that got IVIG plus infliximab. And the way that we defined resistance was kids that got fever 36 hours to 7 days after finishing their first IVIG. And what we found from our clinical trial was that there was no difference between the groups. So there you have on your left-hand side placebo, and on the right infliximab groups. And 11% of kids in both groups had IVIG resistance. So in other words, giving infliximab at the very beginning of a diagnosis of Kawasaki disease in all children that had acute KD did not actually decrease the rate of IVIG resistance. Now you may look at these data and you may say, well then the study was a failure. Well in fact, the reason it got into Lancet was because it was not a failure. And here are some of the reasons why. We actually showed that it was safe to use in Kawasaki disease. That's very important because prolonged use of infliximab has actually been associated with fungal diseases as well as tuberculosis. People are very scared of this particular drug, but they're scared of it in a very different context than KD. They're scared of it with prolonged use with other immunosuppressive agents for things like rheumatoid arthritis. Here is an example of a disease where we're going to give one drug once, and we want to know if it's safe, and in fact it is safe, and it was safe even in infants, where there's a lot of controversy about using this particular drug in Japan. So we're proud to say that in 11 infants less than a year of age who received infliximab, they tolerated it well. So given that we did not show that IVIG resistance rates are any different between the two groups, what is the role of infliximab in Kawasaki disease? And this is why this paper made it into Lancet, was because we showed that it was not only safe, okay, but the data did also suggest some biological and clinical effectiveness. So we had less days of fever, we had reduced inflammation overall in the children that got infliximab, even though treatment resistance did not improve. And so now at Rady Children's Hospital, if we now give infliximab to children that have coronary artery aneurysms and dilations on their first echo, because we know that this is actually a treatment that's been shown to be helpful in those particular areas. The other thing is that we showed that it's safe and it could be a potential rescue therapy for IVIG resistance. So in our center, if your child gets IVIG and then their fever comes back, what we're going to give them as the rescue therapy is going to be infliximab. And based on our data and our manuscript, this is something that other centers are also moving towards in terms of like replacing and giving infliximab rather than giving steroids from the get-go and things of that nature. So we have had an impact globally on the treatment of Kawasaki disease and are very proud of the work. And we're thankful certainly to anyone here whose child was enrolled in either of our infliximab trials. Now, with that in mind, with our initial study, our hope had been that we would be able to have a treatment that worked in all children with Kawasaki disease, that would improve the outcome of all kids. And what we found was that really Kawasaki disease is a very heterogeneous disease. There are children that do beautifully well, probably some that would even do well even without therapy, frankly, but we just can't figure out who those children are from the very beginning. And then there are kids that do very poorly despite getting state-of-the-art therapy. So what we have decided to do is to focus on the kids that are most severely affected. The most current way that we follow these children is by echocardiography. There's no treatment to stop progression of aneurysms in Kawasaki disease. And we really feel that it's our role to change that paradigm, to develop treatments that really we feel are for children with the cardiovascular damage from the very beginning. And infliximab is one of those things, but we need to do more. So how can we do an even better job of treating KD kids with coronary artery damage? So this gets into the world of our, one of our current studies that we're doing right now, one of our current trials, which was just funded by the NIH within the last month. They finally woke up and funded our work with this particular trial. And we're very, very proud of this study. This is actually ongoing. Maybe some of your own kids have been in this. And this is capitalizing on the benefits of the statins. Okay, so the statins are a class of drugs that are very well known for lowering cholesterol, right? So like Lipitor, many of you may have this in your own medicine cabinets, or your parents do, or somebody else does. This is like practically in the water in the US. But the reason that they're so well loved as a medication to be giving in adults is that they have anti-inflammatory properties, they have antioxidant properties, and they also prevent vessel damage and promote vessel healing. Well, if you look at those three things, you say, isn't that what we want in Kawasaki disease, especially with coronary artery damage? So when we got to thinking about KD and the damage that occurs and thinking outside of the box with current treatments, we decided that we would like to capitalize on the statins. Why should it only be adults that have cardiovascular disease that benefit? So we have been repurposing a familiar drug. So Lipitor, we actually have an open study that we designed where we are working with Children's Hospital of Colorado. It's a 2-site study right now. And I'm going to walk through the design of that study. So a child that comes into Rady Children's Hospital that's admitted for Kawasaki disease will go in one of two ways. You're either eligible because your initial echo shows that you have dilated or aneurysms, or you're ineligible because you have normal coronary arteries. This comes with the caveat that because there's not a lot of data about Lipitor being used in children, the FDA will only allow us to enroll children that are age 2 and above in this study. And so children under the age of 2 are not eligible for this study in particular. There's another one for some of the younger kids that I'll talk about in a moment. But if you're 2 and above and you have dilated coronary arteries, you are eligible for this particular study. We, of course, have to walk through the whole informed consent process. For those of you that have been our subjects, you know what that entails. This is probably about an hour of our time sitting down with you, walking through, discussing the risks and benefits of the medication, what is involved in the study. And then from there, this is a study that has multiple dosage levels. And so we are looking at trying to figure out not only if it's safe in our children, but the other objective of this study is what dose should we be using? And no one has ever looked at what dose to use of atorvastatin in a 2-year-old, much less in Kawasaki disease. So that's our charge. And so once a dosage is assigned, and we have specific criteria that I won't walk through, and we have 4 specific dose levels. We're currently on our 2nd dose right now. Then once the child is enrolled, then we're looking at drug safety and tolerability. We're looking at drug levels, so we want to know how that's handled by the body. And then we're measuring biological activity, getting back to the anti-inflammatory, antioxidant properties of atorvastatin in general. We want to be able to look at that within our patients. And as I said, we're now funded by the NIH for doing that, although we're always looking for more funding. So that's our Lipitor atorvastatin study that's going on right now. Okay, now we're going to switch gears because, wow, 2 and above. Unfortunately, I know that most of our kids that get really bad heart disease are below age 2. So we felt very strongly that we needed to look for other agents that are used for other diseases in younger children. And that brought us to this family, the IL-1 family, the interleukin-1 family. This is a family of 11 proteins, okay, that their main function is to control inflammation, inflammatory reactions to what's called pathogen-associated molecular patterns, which is basically what your body reacts to when it gets what it calls a foreign invader, something that it's not used to seeing. And you can see at the very top there, on your left-hand side there, at the very top you have the IL-1 beta family, and you see all these molecules, some floating outside, some attached to the cell membrane. And then this whole cascade of stuff that happens, and then it leads down into the nucleus of the cell. And in that bottom box there are some other inflammatory markers, including TNF-alpha that we just talked about a moment ago for the infliximab. So infliximab targets specifically TNF-alpha. It turns out that drugs that have an effect on IL-1 beta, since IL-1 is actually upstream of all these inflammatory markers in the body, that it inhibits actually more than just TNF-alpha. Okay, so we're looking at an upstream area now, and we're asking if we actually change how the body, or if we interfere with the IL-1 response in the body, can we interfere with the progression of aneurysms and dilation? So this rationale for doing this work, I'm going to show you a case in a human. I'll talk to you about a personal case of mine. But this work, in part, is also from some mouse data. This is by a group up at Cedars-Sinai, Moshe Rady. And here's a rationale for the IL-1 blockade in KD. So there is a mouse model of vasculitis. Okay, so these mice get injected with a form of crushed up yeast through their belly that then causes vascular inflammation of their coronary arteries that seems to look like Kawasaki disease. The mice don't get red eyes or red swollen hands and feet, but they do seem to get the vascular disease. So this is used as a mouse model for Kawasaki disease. And it turns out that the WT there means wild type. That's a regular mouse that gets this injection in the belly and then gets this severe amount of inflammation in the vascular wall. These are all inflammatory cells here. Okay. Now, in a mouse where you knock out the IL-1 response, you don't allow the body to have its natural response with IL-1, what you see is a much more normal vessel. This is what a vessel should look like. And so this is a compelling argument that blockade of the IL-1 cascade can actually be part of hopefully interfering with the inflammation that happens in the coronary vasculature in Kawasaki disease. Now I'm going to talk about an anecdotal case. So this is a case actually from the Netherlands by one of our KD collaborators. And they had a child with Kawasaki disease and aneurysms that was refractory to multiple therapies. I won't go into all the details, but this kid had pretty much gotten everything that could be given in KD and a lot of it, and had still some very large aneurysms. And they gave anakinra, and the aneurysms appeared to have remodeled, which is extraordinary. Granted, this is an N of 1, okay? And then when they took off the medication, they had him on a medication called anakinra that I'll talk about more in a minute. When they took him off the anakinra, the aneurysms worsened when the medication was abruptly discontinued. So we'd heard about this case. This was published last year in 2013. We knew of Moshe Rady's work with the mice. There'd been a lot of discussion in the community about anakinra. And I had been working on a protocol to be able to use anakinra in our Kawasaki disease patients. I'm happy to tell you all that we now hold what's called the investigational new drug. And that is what's called an IND. An IND is what the FDA grants an investigator when they feel that you have justified the use of a medication in a particular population. So anakinra is currently on the market for some very rare diseases, some rare inflammatory diseases in children. It is not approved for Kawasaki disease. This is certainly what they call an off-label use. So we applied to the FDA in order to be able to be granted the use of anakinra, and they granted us the IND. So we have an ongoing protocol for the use of anakinra. Now, before our protocol even opened and our IRB approval was in, I was faced with the situation where I had a child that had developing aneurysms that had other infectious diseases at the same time she had her KD, and the child had respiratory syncytial virus and had a respiratory illness as well as having a urinary tract infection. Infliximab would have been my first choice for that child, given that I knew that she had coronary artery disease. The problem was that infliximab lasts anywhere in the body from 5 to 10 weeks, and I didn't really want to give an immunosuppressive drug to a child that I knew had other infections plus severe KD. So I reached into my armamentarium, called my colleague and KD queen and mother, Jane Burns, and said, okay, what are we going to do for this kid? Because I can't just sit here and wait. And said, let's just give anakinra and see what happens. So that's the only child that's ever been treated in North America. We were able to control inflammation of that child with a brief course of anakinra. As they left the hospital and we stopped the anakinra, we gave them a dose of infliximab because I'm not looking to prove that anakinra works with that child. I just want that child to get better. And over time, those aneurysms have been improving. Now, would that have happened in the absence of anakinra? I will never know. But frankly, as a provider of medical care, that doesn't matter to me. My goal is to make every child that presents better. But for me to be able to know whether anakinra is the right drug or not to give, then we're really looking at a clinical trial. So we have designed that clinical trial for anakinra. It's currently funded by some local grants from the hospital, from Rady Children's Hospital, as well as the university. We will be turning that into an FDA orphan drug grant in February. And that is a study that's currently open. We've been given permission by the FDA to use anakinra in children 8 months and above at the moment. So we are still looking for other drugs that we can use in even younger children. And we will be treating those kids for 6 weeks. So this work really does take a village of people to do. In my mind, it can't be done without families and patients putting their trust in us. Here I am saying, by the way, your child has this, disease that you've probably never heard of, has bad heart disease, and oh, by the way, can you, trust in us to give your child this particular drug for another 6 weeks? But we're incredibly grateful to all of you for entrusting your children to us. And really, my goal as I stand up here is to have an impact on all children so that we really reduce the morbidity and mortality of Kawasaki disease globally. And that's what these efforts are towards. So I appreciate any follow-up questions that you may have during our break time, and I'll be certainly around for questions afterwards. Thank you.
So Dr. Daniels is going to be our next speaker. Dr. Daniels and Dr. Gordon are 2 members of our adult Kawasaki disease team. So we'll be switching now to an older age group. We've heard about the babies. Now we're going to hear about adults. Thank you, Lori.
Thanks. Hi, everyone. It's good to be here today, and welcome to those watching the online stream as well. I'm going to be talking about some of the results of our San Diego adult KD collaborative study, which is a study that we started about 5 or 6 years ago now to better learn what happens to KD patients as they grow up. As Jane mentioned earlier in one of her talks, as Dr. Burns mentioned earlier in one of her talks, this is, as far as we know, the only systematic study of this in the country and possibly in the world. In order to be able to tell all of you what are the long-term consequences, hopefully none, of Kawasaki disease, we need to study in a systematic fashion. And that's what we're trying to do. So the overall goal of the study is to answer this question: what does the future hold for individuals with a history of Kawasaki disease? Can everyone hear me okay in the back? Okay. How will having had Kawasaki disease affect future cardiovascular health, as well as other problems? Is there any risk for individuals who had normal echocardiograms during the acute illness? And can we predict who is at risk, if anyone, using simple, non-invasive tests? So that when you come for follow-up, we know what to tell you. We know how to keep you healthy, and you know what to expect. So this is from the San Diego, this was in the San Diego paper. I guess they borrowed it from the Roanoke Times. But this woman is worried about the effect on her unborn child from the sound of jackhammers. And as you can see, what's she doing here? She's smoking. The point of this slide is that there's some things we should worry about, and there's some things that maybe we don't have to worry about as much, right? And part of what we need to do is focus on what we can control and what we know is healthy. And we'll learn more. We're always learning more about how to keep KD kids and young adults healthy. But don't forget the obvious things either. But there we go. But a lot of KD parents whom I've gotten to talk to over the years, I think probably feel somewhat like this man. I mean, they don't know something's coming, but they just feel like they're about to get hit with something, right? In this case, the man is right, but I don't know that that's true for all of us, and we want to get more information about that. I'm having trouble advancing slides here. Is there any? Okay. So there's a lot of components to our study. Now, we're not doing all of these at this time, but when we started the study for the first 4 or 5 years, while we had funding from the American Heart Association and the Macklin Foundation, we were able to do a lot of this, and we're still doing some of it. So one of the most important parts of this study is the health questionnaire. And it's a comprehensive questionnaire about every risk factor, every health outcome that one could possibly think of, so that we can know exactly how your health is and how it progresses over the years. We follow up with every 2-year follow-up questionnaires. And incidentally, we also enroll healthy controls, which I'll talk about in a minute. So the questionnaire is very important. At baseline, we also get electrocardiograms. Typically, the individuals, if they're local, will see a doctor and have a physical exam. We do some blood work so that we can measure biomarkers, kind of like Dr. Tremblay was talking before, because in addition to using blood tests for a diagnostic test, we can use it to predict future risk, we're hoping. We have gotten echocardiograms on folks, coronary artery calcium score, which Dr. Gordon is going to talk more about later and has some really interesting results. And then we've been doing some newer things like testing the function of blood vessels, which I'll talk to you about, measuring the carotid arteries health. If it's indicated, if someone has symptoms, we've been including stress tests. And sometimes if there's aneurysms, cardiac MRIs as well. And then I mentioned the longitudinal follow-up. That means we contact people after a couple of years so that they can update us on their health history. And that's how we learn over the long term about what's going on. So we've enrolled 3 different categories of individuals. So first we just decided to call them cohort 1. It's young adults, and typically to enter the study, you have to be at least 15. Young adults with a history of Kawasaki disease who've been followed here with Dr. Burns or Dr. Tremblay at Rady Children's Hospital since childhood. We've known about you since then. Your local San Diegan, cohort 1. Those are the folks who give us the best information in terms of the natural history of disease, because we know all of your data from the get-go. But we also enroll another important group of individuals called cohort 2. Those are folks with a history of Kawasaki disease who found us somehow, either through the KD Foundation, through an internet search, whatever. They heard about our study and they want to join. That's called cohort 2. We haven't been following them since the get-go, so not everyone in that group might be fully representative of the natural history because they found us. And sometimes individuals who contact us are the ones who've had more problems with their KD. But it's an important group for us to learn from because they have a lot of information to tell us that we can gather. And finally, the third important group is the control group. Dr. Tremblay mentioned it, and in any scientific study, if you're going to make comparisons, it's important to have a control group so that you can say, okay, this % of the healthy non-Kawasaki disease population gets psoriasis, maybe 2%. But we see it in 5%, for example, of the kids with a history of Kawasaki disease. You can make that comparison because some people without a history of Kawasaki disease will have these things too. And that enables us to make comparisons. So if you're someone who hasn't had Kawasaki disease, but you're 15 or older, we'd still love to have you join our study so that we can use you as a healthy control, okay? You're just as important to us. So here is an updated table of how we're doing in terms of enrollment in the study. And the total number of enrolled individuals so far, we have 67 cohort 1, so local San Diego folks who've been followed here from the get-go. We have 104 from cohort 2, and we have 139 healthy controls. So closely balanced, but as you can see, we still need some more healthy controls, and we try to age match them to our Kawasaki patients. Let me talk to you about a couple of the components that we're doing or that we have done and we hope to keep doing as we get more grant funding. One is measurement of the carotid artery with the carotid ultrasound. So it's a non-invasive test, kind of like an echocardiogram, but instead of putting the probe here, we put it up here. And what we do is you can look at the carotid artery and how thick the wall is and also how much the wall stretches with each beat of the heart. we're trying to learn whether individuals with a history of Kawasaki disease have abnormal thickness in their carotid arteries, because we know that Kawasaki disease affects the blood vessels. And at least in general cardiology, when we're looking for typical atherosclerosis, the typical disease process by which Americans and individuals get heart attacks and strokes, we know that thickening of the carotid artery, the intima, the IMT, the intima media thickness, is associated with an increased risk of heart attacks and strokes. But we also know that the pathology of Kawasaki disease, what goes wrong in the arteries of Kawasaki disease patients, is not the same as what you get from typical atherosclerosis. It's not the same thing going on in there. What we don't know is whether Kawasaki disease patients and individuals will get thickening of these coronary arteries, carotid arteries, and whether that can predict future risk. So to start to look at this, we measured the thickness in the carotids, and we compared the thickness among our cohorts, among our control patients, and then the KD patients, based on whether they always had normal coronary arteries, whether they had transiently dilated coronary arteries, or whether they had a history of aneurysms. And the good news here, and it is good news, is that there was essentially no statistical difference in the thickness of the carotid arteries between Kawasaki, kids, young adults with a history of Kawasaki disease, and normal healthy controls who never had Kawasaki disease. So that's good news. We're not seeing abnormal thickening of the carotid artery. The next way, though, that we looked at the carotid artery is something called carotid artery wall strain. So it sounds like a bad word. Strain sounds like something bad. But in the setting of the carotid artery, strain is actually a good thing. So the more your carotid artery strain score is, the more it's able to stretch with each beat and respond appropriately. You can think of it that way. And we found an interesting, a kind of unexpected finding here. And for those of you who were here last year, there's a common theme. I showed this, a similar result, but with a different test that we'll get to. What we found is that the KD young adults who had a history of transient dilation of their coronary arteries had actually what we would consider a healthier strain pattern in their carotid arteries compared to the other Kawasaki disease kids, and even compared to the normal healthy controls. So that's a surprise, because you know, if you're a parent of a kid who had transient dilation, one question that we get a lot is, you know, are they at increased risk, like kids with aneurysms or things like that? Are they going to get aneurysms? What's going on here? Why did they only have transient dilation? Well, we don't know the answer to that yet, but this is a reassuring result that maybe the reason that they were transiently dilated and didn't go on to get aneurysms is that something in their blood vessels is a little bit healthier and helps it bounce back to normal. This is a hypothesis right now. As you can see in this slide especially, there were only 5 in that category. And we're really trying hard to enroll more young adults in all of these categories, but especially this one, so that we can see if this is just an accident of the data or whether it's going to hold true when we look at other things. But you're going to see that theme in some of my data here. These prior dilated coronary artery individuals had this healthier strain pattern. Another test that we looked at is called the ankle brachial index. It sounds complex, but it's one of the most simple tests we do. We basically just take the blood pressure in the arm and compare it to the blood pressure in the leg. Now, this is something that's done in older adults all the time in the cardiology office to assess for what we call peripheral vascular disease, hardening of the arteries in the legs. We didn't know whether we would see anything like that in our Kawasaki disease survivors. And when we looked at the results, it was also essentially good news. There was no statistical difference between control patients and individuals with Kawasaki disease, especially in the normal or dilated group. The numbers are small. There were a couple of individuals with aneurysms who had a slightly abnormal pattern, but at the same time, those folks who in that group were also a little bit older and might have just had the abnormal pattern because of advancing age, and they would have gotten that anyway. But overall, no significant difference there. When we look at it on a continuous scale, basically it shows the same thing. no significant difference between those with aneurysms, dilated coronary arteries, and normal status. So that was reassuring. Finally, we did a test called the EndoPat. This is a test that checks endothelial cell function. What are endothelial cells? Endothelial cells are the little cells that line your blood vessels. And the lining of your blood vessels are actually really important for your cardiovascular health because they send out all these little chemicals to help the blood vessels dilate, get bigger in response to stress and exercise, and to shrink back to normal again. Now, once again, we know in the setting of typical heart attacks that people get with age, we know that in that setting, endothelial cell function is worse. As you get older, it gets a little bit worse, and if you have your typical heart disease, it gets worse. But what we didn't know is if Kawasaki disease affects endothelial cell function, and whether it's different in folks with a history of aneurysms or transient dilation. So just briefly, I'm not going to get into the real science of how this thing works, but it's a non-invasive test. It doesn't hurt. We're able to do it on these young adults with no problem. You have a little probe on your fingertip, and it's measuring basically the pulsatile volume changes, the microscopic volume changes in your finger with each beat. And what you do is we take a blood pressure cuff, and for 5 minutes, we pump up that blood pressure cuff pretty high, not so that it hurts, but so that it occludes most flow to the hand. And then after 5 minutes, we release the cuff. And what happens is that the blood flows back into the hand. And a normal response, if these endothelial cells are healthy, is that the blood flow as it rushes back in is higher than before to make up for that lost time. And you can see that here in the test arm. Here's the blood flow through the arm, and then you pump up the blood pressure cuff, there's no flow, and then when you release it, you get even more flow than before. That's a normal response. This is the control arm where you don't do anything and it stays about the same the whole time. So this is an example of a tracing that we get. Here's someone pre-occlusion, they've got good flow, you occlude it with the blood pressure cuff, there's no flow, and post-occlusion, you get an increased flow. That's a normal response, and this would be an abnormal response. The flow resumes, but it's not better than it was before, and that signifies less healthy endothelial cells, if you can see that difference. So what did we find in our Kawasaki disease folks? Well, it's a pattern like what I showed you for the carotid arteries. It didn't, in general, folks with a history of Kawasaki disease didn't have any abnormalities. It was good news. They were the same as healthy controls. And the one exception was that individuals with this history of dilated, transiently dilated blood vessels had a supernormal, a healthier response compared to healthy controls. And you can see that in yellow. And again, that was unexpected because we thought, if anything, folks with dilation of their arteries, it would imply worse function of their endothelial cells. But maybe they have these healthier blood vessels, and that's why it's able to resist the damage from Kawasaki disease, maybe. And as you see in this test, we're up to 16, so we have more numbers here. Still, what I would consider preliminary results and a small number. So we're still trying to get more folks into this group as they age up and are able to do it. But healthier endothelial cell function in these individuals. And this is just another way of looking at the same data. It's the flow in that fingertip over time after you release the blood pressure cuff. And yellow are these dilated, transiently dilated folks. And you can see that that flow increases more quickly and drops back down to normal more slowly. So they get a better, more vigorous response. Finally, from the same blood test, we can also get a measure of something called the arterial stiffness, how stiff those blood vessels are. And in normal individuals, again, this is something that as we get older, our vessels get stiffer, okay? And we tend to think of that as it's normal, a normal sign of aging, but it's not necessarily a sign of health. The younger you are, the less stiff your vessels are, and we can measure that with this EndoPat. And when we measured it in our Kawasaki disease patients, I'm sounding like a broken record now, but we saw the exact same pattern. The only significant difference was in the transiently dilated patients who had less stiff arteries than their healthy control counterparts. So again, possibly a healthier response there. And now we've seen it in 3 distinct tests. So we're still getting more data. We're still trying to enroll individuals. Some of you who've already enrolled, we thank you for that. You may be hearing from us soon if it's been 2 years since you enrolled, because we've started our 2-year follow-up, and you might be getting a questionnaire just so we can check in on you. Expect an email. We've been trying to do most of this with SurveyMonkey links, and that saves everyone time, and you can answer the questions on your own time rather than having to do it by phone. So thank you for doing that. In the meantime, while we wait for all these results, what can you do? Now, Dr. Burns wanted me to make sure to remind everyone that the same risk factors that cause heart attacks don't cause problems in Kawasaki disease, but I, as an adult cardiologist, still think we need to be heart healthy, so we don't, you know, one of my mentors used to say, just because you have ticks doesn't mean you can't have lice, right? I don't particularly like that expression, but I think the point is, let's not give ourselves another disease. So let's follow a heart-healthy diet, not like something that my patient nicely tweeted to me, which almost gave me a heart attack. Let's all keep up the heart-healthy exercise. Let's do the things that we know how to do. And I don't mean to preach from the choir here, but here's my colleague cardiologist at the American College of Cardiology, and we're not, no one's perfect, but we can at least try, right? So for all of us, we should make sure you have your blood pressure under control, monitor your cholesterol, and this person, she knows how to control stress. But before I make all of you look like that, I think I will finish and say thank you for your attention. A key summary point about what we know so far, and Dr. Gordon is going to add more to this with his slides, but we think that patients with normal echocardiograms throughout their initial course may not need long-term cardiology follow-up, although before we determine that, we recommend probably a CT calcium score once they're 10 years out from their Kawasaki disease. Dr. Gordon's going to talk more about that. But for patients with any coronary artery abnormality, and for now we're including the transiently dilated ones in that group, that might change if we get more information that confirms these findings. We think they need follow-up for life by a cardiologist knowledgeable about the vascular changes with Kawasaki disease. So if you want to join the study, we're still enrolling, as I mentioned, at least for questionnaires for sure, and as we get more funding for the other components, there's a table in the atrium where you can sign up, and if you're a healthy control, you can sign up as well. So thank you to all the KD patients and families, all the control subjects, our funding agencies, and to my colleagues, of course. Thanks for your attention.
So thank you, Lori. And Lori's going to stick around for questions after we hear from Dr. Gordon, so it'll be more about adult Kawasaki disease issues. And our adult Kawasaki team is sub-specialized into areas where they have particular expertise. So Dr. Daniel's expertise is in biomarkers and these non-invasive measures of function. Dr. Gordon is more the plumber. He's the guy who goes in and fixes the blocked arteries and evaluates problems after people have had a heart attack. So I'm going to turn it over to him, and then after that, we're going to take questions the same way we did in the previous format. Thank you. Dr. Gordon.
Well, good morning. Can you hear me? Is this working? No? Maybe I'll hold it. So, once again, good morning. As Dr. Burns was telling you, I'm an adult cardiologist, so I think most of you here have young children and haven't had to go see the adult cardiologist. But I'm here to tell you a little bit about the future. For most of you, your children are going to grow up and they're going to be absolutely fine. And Matthew, maybe we need slides. Yep, there we go. But what we've lost actually is a picture of Dr. Kawasaki. And what I wanted to show you is a young Dr. Kawasaki in Tokyo. And I like to show this picture, primarily because he did something extraordinary then. He had eyes that could see a new disease that others missed. What he didn't really know was how things were going to go in the long term for these children. And he assumed that things were going to be fine. I'm going to show you one patient of mine who I met because he was having a heart attack. So a 32-year-old man with no traditional risk factors for coronary artery disease presents in our emergency room. And what you can see here are, and I know none of you are used to looking at arteries, but these are what we call giant aneurysms. They're about 3 or 4 times the size of your arteries or normal arteries. And here's the problem. His right coronary artery, so the artery that goes around and goes underneath the heart, is totally filled up with blood clot, and his heart's not happy about that. So that's why he came to the hospital. So what do we do? It turns out we can generally fix these things. So we put a wire through the blood clot, and there's a balloon here, which basically allowed us to improve the flow. We also use a number of different anticoagulant medicines to dissolve the clot and prevent the clot from coming back. Now you can see an artery that you couldn't see before. So his heart's much happier at this point. And for 2 months, we put him on aspirin, a medicine called Plavix, and Coumadin. And if I can advance this, I'll show you that the blood clot has now entirely resolved. And so you can now see but you can appreciate now how big this artery was. And here's his heart, which is actually working quite well. The artery supplies the underside of the heart, and you can see he got to the hospital early enough. We got the artery opened up, and he's actually doing fine. So this was about 7 years ago. I still see him in the office, and he's doing well. So so this is worst case, the heart attack. This is what we'd like to prevent. Now, let me show you another case. This was a patient who had Kawasaki disease at age 3, and he had dilated arteries that normalized by echo. So he was really thought to be okay by his pediatricians, was reassured, and sent on his way. And he was athletic, he was a runner, and when he was 32, there's really nothing magical about the age of 32, but my 2 patients have that. He developed what we call unstable angina. When he was running, he would get discomfort in his chest, and when he rested, it went away. So he went to see the cardiologist, got a stress echo, which was abnormal, and they did an angiogram. And so once again, what you see here, this is a very tight blockage in the artery that supplies about 2/3 of the heart muscle. So a very important blockage in a very important artery. You don't really see any big aneurysm here, but what you see are, I think, is the consequence of an injury in childhood. And this is part of the reason that we're not so sure that you can be confident discharging patients who have, quote, normalized from care. And we'll talk in just a minute about what we recommend doing. Now, Dr. Daniels alluded to this, and I think this is a graph I'll explain to you, but as part of our adult collaborative study, one of the tests we did is called a calcium coronary artery score. And this is a very simple test that takes less than 5 minutes to do, very little radiation. And what it does is it allows the radiologist to quantitate how much calcium there is in a coronary artery. Okay, so 0 is a good number. If you don't have any calcium, you have a normal artery. and a very good prognosis. And so we took all of our patients, and I think there are about 160. And what you can see is that up to about 10 years, we didn't see any calcium at all. So you see this blank spot here. But our patients who had aneurysms, all these blue dots, after 10 years began to show a calcium signal. So what this, I think, teaches us is that we now have a tool that will allow us to find patients who don't know they have a problem, or who were told they were fine. And if they have calcium, that may not be true, or it's not likely to be as true. So we now have a test for figuring out who these patients might be. And this is just a model in the CT scanner. Again, low radiation, simple, takes a few minutes only. This is the calcium score on this patient I just showed you, who had unstable angina at age 32. And so if we had done this test, we might have picked up his coronary disease or his important narrowing earlier. Fortunately, he didn't have a problem, but he might have. Now, having learned about the calcium score, in Dr. Burns' clinic, the uncle of one of her patients showed up. And he said, well, I had Kawasaki disease when I was a child, and I was fine, or I am fine, but I'm happy to be in your study. So we did a calcium score test on him, and guess what? He had important calcium that these arrows are pointing to. And this was an abnormal study, so we looked farther, and what we found was that he had a 7.1 millimeter aneurysm that was unsuspected in his what's called the circumflex artery to the side of the heart. And that's actually hard for the echocardiographers to see. So this is easy for them to miss. But this calcium test allowed us to find what could have been a heart attack in waiting, and allowed us to treat him properly and potentially prevent a heart attack. So let's see. Okay, there we go. So what do we propose to do, or how do we manage these young adults who have had Kawasaki disease? Well, if there's no abnormality, generally it's yippee. Everything really is very, very likely to be absolutely fine. So we do in young adulthood at some point we would recommend a CT calcium score and an echo. If the calcium score is 0, the echo is normal, then I think it's very likely that person or that child or that young adult is now fine. We reassure them there's no medicine needed. They should stay away from the cheeseburgers and all of that we just heard about. But they don't really need to be followed from a cardiac standpoint. For patients who have transient dilatation in childhood, we would do the same test, a CT calcium score and an echo. And if they're absolutely normal, they should be, we would reassure them. And I think no medication is actually warranted. Whether or not we should discharge them from care, I don't think anybody really knows. And that's part of what we hope to learn from our collaborative callback study. And so we'll see. But we do anecdotally have patients like the second one I showed you who was thought to be fine, and he wasn't. So if there was evidence for injury, we think those patients should be followed long-term. And small to medium-sized aneurysms, now we're getting into patients who we know had some important injury. We, again, do the same test, CT angiogram. We do what's called a stress echo. I'll show you a picture of someone undergoing that test in a minute, but it's essentially just an exercise treadmill test where we use ultrasound to look at the ventricle and see how well it pumps. And if that functional test is normal, then there's not likely to be a blockage. And the echo allows us to look at the heart and see how it's doing. We do an electrocardiogram, and we ask them to come back for regular follow-up, perhaps once a year. These patients are treated with aspirin. This illustrates the stress echo. You can see the treadmill. This panel over here shows you what the echocardiographers are looking at. And basically what they hope to see is that after exercise, the heart is pumping stronger, that all of the walls are moving normally. If one of the walls of the heart is not pumping as well, that reflects a lack of blood flow and a problem. And that would be a positive test. Some adults, and I think not most young adults can exercise, but if one can't exercise, we can do pharmacologic stress testing, and we can use nuclear imaging to do that. And what we would prefer, if we can, is we'd prefer to avoid radiation from this kind of test, but it's very useful for those who can't exercise. Now, how about for patients like the first one I showed you? So for patients who have what we call giant or quite large aneurysms, those patients will need lifelong follow-up, and that group will have more problems than the less severely affected patients. So we do stress echoes on a regular basis, and obviously if the patient has symptoms or has an abnormal test, then we would proceed to angiography and fix a blockage if that's what happened. So what are the problems that occur downstream or as time passes? You can get what's called a stenosis or narrowing of the artery, or you can get thrombosis or blood clots in the aneurysms. So our therapies are designed to prevent those problems and to manage them should they occur. So we would also do an electrocardiogram, treat these patients with aspirin. Most of you, I'm sure, are familiar with warfarin or Coumadin. It's an anticoagulant. And we now have some new, what are called novel anticoagulants that allow us to prevent blood clots in the heart without doing routine blood testing. So, and we will actually formally study those as soon as we have funding. But at this time, there's actually no evidence-based medicine that this is an appropriate therapy. So I think what I want to leave you with is that with research, with the knowledge that we're going to gain from the studies that are in progress, the future is quite bright. I'll leave it there, and we'll open it up for questions.
Pass once again your index cards, and we'll get some people to walk up and down the aisles and collect them. If anybody needs an index card, raise your hand, and we'll have ask Dr. Daniels and Dr. Gordon to come up, and we'll sift through the questions and answer them. While those are being collected, there were a few questions that we didn't get to from the last session, and so that's yours. And so I just wanted to answer those while we're collecting these other ones. There was some confusion about what we were talking about the exit visit from our pediatric clinic before we make the decision as to whether or not patients need to be passed on to Dr. Gordon or Dr. Daniels. And we were recommending an echocardiogram, fasting lipids, and people were wondering, why are we doing a fasting lipid panel? And the answer, I hope, was addressed by Dr. Daniels. It's not that there's any relationship at all. In fact, we've just published data using the blood from your children that there is no relationship between atherosclerosis, the typical kind of heart disease, and the Kawasaki kind of damage to the blood vessels. But just because you had Kawasaki disease, it doesn't necessarily protect you from also having the typical kind of heart disease. So this is just a precaution. We don't want to give people two diseases. You already have one problem, which was that your child or the young adult had Kawasaki disease. And we don't want to superimpose on top of that also the regular kind of heart disease that is related to lipids being deposited into the wall of the artery. So that's why we do the fasting lipid panel. We're looking for children who have been inherited just by bad luck, not because of an association with their Kawasaki disease, but by bad luck. They've inherited a not so great way of handling the lipids in their diet. We want to identify that genetic risk and then counsel people about a heart healthy diet and exercise and all the things that Dr. Daniels mentioned. So two different problems, but they could be so superimposed, and we don't want to give the coronary arteries two hits. We mentioned blood pressure is always part of every physical examination that we do. The CT calcium score was already mentioned. So when you're ready, Lori, go ahead and take over. There was also a question from someone who said you've recommended that our child be followed up every year, and that must be because your child had some evidence of damage to the coronary arteries. So for people who had children who had evidence of damage, and when they become adults as well, we recommend a yearly follow-up visit with the testing being individualized to whatever the specific problem is with that patient. So those were the reasons. There's a question here about the development of arthritis after Kawasaki disease. We have seen that. We have some patients who have developed different forms of rheumatoid arthritis that can happen in young people. We don't know if that's true and unrelated, or whether it has to do something with the differences in the immune system in these patients. That's why Dr. Daniels is doing the follow-up study, and please participate, and when your children age up, have them participate. It's the only way that we're going to be able to answer those questions. And I'm going to turn it over to you guys to answer your questions now.
Okay, this is a question that I think must have been written by a physician in the audience, or at least someone very well educated with echocardiograms. It says, has there been any recent research regarding incomplete relaxation of the heart muscle in older Kawasaki disease patients? So one of the things we can look at when we do echocardiograms is how well the heart relaxes. And that's related to a lot of factors, but we know that some individuals with Kawasaki disease can get stiffening of the heart muscle. So that's some data that we're excited to look at as we get more echocardiograms and as we're able to process that data. The data on that has been mixed. There's certainly a small subset of individuals with a history of Kawasaki disease who as they get older might have a more accelerated stiffening of the heart muscle. This happens normally with age as well, but the jury is still out, I would say, on the answer to that. This question, is my son at higher risk for heart disease since my father had angina and COPD and a heart attack? So that's not a Kawasaki disease question, but the answer is possibly yes. If you have a family history of heart disease, then you're at risk, and it's more important than ever to follow a heart-healthy lifestyle and not smoke, make sure you exercise, etc. And finally, as an adult with KD and aneurysms, how frequently would you recommend to have calcium test, stress test? I'll give my opinion on this, and John, if you want to weigh in as well, feel free. I don't think regular calcium tests or stress tests like clockwork are indicated. I think once you know you have aneurysms, the calcium test isn't really useful at all anymore. What is important is, number one, if you're getting symptoms, chest discomfort. Number two, if you're noticing a decline in your exercise tolerance. Those would be the types of things, in my opinion, that would trigger a stress test. We don't do it routinely if you're able to exercise and feeling great and nothing's going on.
No, absolutely. I just want to emphasize that the calcium test is really, and actually all tests we do if we want to answer a question. If there's in the case of the calcium score, it's really useful for helping us to pick up patients who we're unsure about. But let's say once the test is positive, you're done. You don't need to repeat that. You know there's a problem that you then investigate. But and with respect to stress testing, no one really does know what the frequency of testing needs to be, but it certainly doesn't. You know, we had, I think, once a year on some of these, but generally patients who need a stress test are those who now have angina or have exertion-related chest discomfort. And it helps us to learn how severe the coronary disease is, or also helps us to know if the chest discomfort is from the heart, because there are many reasons for chest pain. This is where this is why you need a doctor once in probably annually if you have had aneurysms, just to talk to, and certainly if you develop symptoms, you need to go and see the doctor. I have 3 questions I'll go through. The first is, if my son has an aortic aneurysm, what symptoms would you look for? What warning symptoms? And these are generally silent, actually. Aortic aneurysms, if they become quite large, can tear. So what we look for is a progressive dilatation of the aorta. So this is a situation that needs to be followed probably annually with an echo or a CT to assess how big the aorta is. And if it's beyond a certain size, it needs to be repaired. So if there are symptoms, it's not too late, but it would be a bad sign and you need to go to the hospital. And it says, is it okay if my son took ADHD medicine? I think this is not going to have any effect on the coronary arteries. There are some valvular issues with the medicines for ADHD. So next question. Are there any new surgeries to reverse damage caused by large aneurysms? And the answer is yes. Fortunately, so we just had a patient who had a progressively dilating aneurysm of the left anterior descending artery. And that's unusual, so it required an unusually creative operation where the blood vessel was bypassed, part of it was trimmed, and that's unusual. I don't really expect to see another one of those cases for a while. The types of surgical interventions that we do are done generally because there's a progressive stenosis, and that's really this type of operation can be done by a well-trained cardiovascular surgeon that does bypass, but that's not necessarily new. So third question, how long do KD children post-bypass survive? And how long does a bypass typically last? So the answer to that is the arterial grafts that are typically used after bypass will last indefinitely. And if we also use vein grafts from the legs, the veins weren't really meant to be arteries, and they will degenerate over time. And what we've learned from the patients that we've had who are bypassed, say in their 20s, is that these bypasses actually typically do much better than our older atherosclerotic patients, because there isn't they don't have a problem because they're smokers or they have diabetes or whatever. They had Kawasaki disease, and they have tended to do quite well. So how long do they last? We still don't really have long-term follow-up. The largest study from Japan has about 20 years of follow-up. So if we're following patients who had Kawasaki disease at age 2 and they're now in their 20s, we really don't have data from when they're 40 or 60. So that's part of what we're trying to do. We're trying to learn trying to take the best care that we can of these arteries so that they do last. So that's all I have. You have that?
We have a couple more questions from people, and then I think we'll wrap it up. And of course, we're all here individually if you want to come up in person and ask us questions. But someone wanted to know if the wind really comes from China. What about the incidence of Kawasaki disease in the region where we think these particles come from? And that's a great question, and we've thought long and hard about that, because actually the rate within that specific region of China is very low. It's about 5 per 100,000 children less than 5 years of age, compared to 40 per 100,000 here and 290 per 100,000 in Japan. And so the climate scientists and chemists that we work with are also thinking about the idea that maybe when the particles are created in the source region, they actually go up and travel through clouds which contain water, and the chemistry of water on a particle actually turns it into something that's bad for you, as opposed to it being something bad for you in the place where the particle initially was generated. So obviously this is a lot of hand waving, trying to explain pieces of data that are real observations, and we're trying to make all the pieces of the puzzle fit together. So I hope when I get up here next year, I'm going to have much clearer answers about that for you. And there was another question about the age of the oldest Kawasaki disease patient. And actually, as far as we're aware, as far as Dr. Kawasaki or Dr. Yanagawa, the epidemiologist in Japan, are aware, the oldest patient was actually a gentleman who was cared for in John's practice, who we think had his Kawasaki disease in 1945 in Japan. He recently died from heart failure. He had very, very severe damage to his heart. But we think this gentleman, born in 1945, was, until very recently, the longest surviving person with Kawasaki disease. So again, emphasizing that we don't have good long-term follow-up. Some of you may also be aware that there was a disease that we think looks indistinguishable from Kawasaki disease that was very rare in Western Europe, South America, and in the United States, where there are cases reported that go all the way back to the late 19th century. These were invariably fatal diseases in very young babies. And whether that rare disease called periarteritis nodosa of infancy, whether or not that disease and Kawasaki disease will ultimately be found to be the same, and we'll have the concept that Kawasaki disease in little babies was very rare in the Western world, but not unheard of, but definitely new in Japan. We're still sorting that out, and we may not know the answer to that until we actually know the cause of this disease. Do you have some more?
So I have one more question. This says, multiple giant aneurysms underwent bypass surgery, treated with Coumadin and aspirin. But my son is stubborn with aspirin and takes Coumadin, no problem. So why the need for aspirin? So I think that's a good question. And I think what may be a surprise to some of you is that a lot of what we do in with respect to our Kawasaki patients has to do with traditional approaches to atherosclerotic disease. So aspirin has not actually ever been studied in any systematic way. So we don't really know if it's required or not. It's inexpensive. It prevents blood from clotting. It's, I think, probably a medicine that we use traditionally. So is there really a need for aspirin after bypass? Our adult patients with atherosclerosis do all take it, and there is evidence that it helps them. So I think if it's tolerated, it's inexpensive, and not likely to be a big downside.
There's a question about the timing of treatment and whether the timing of treatment affects the development of dilation or aneurysms. Clearly, the earlier that we treat, the better. And we definitely want to catch kids as early as possible. And sometimes that's just not possible, given when children are identified. There are other factors that take a very important role as well. And we know that from a very recent study that we're going to be presenting in Hawaii, that we looked at children less than 6 months of age that have Kawasaki disease, and they actually have a much larger aneurysm rate than children older than 6 months of age, despite being treated in a timely fashion and with the proper treatment. This is within our own center here. They have a much higher aneurysm rate. So there's more than just timing, but certainly the earlier that you treat, the better that is for our patients. Most coronary artery abnormalities, both that we've seen in our center as well as from Children's Hospital of Colorado, there's been a study there too. Most kids that are going to have any heart damage have it within the first 10 days of illness. So it's there's some misconception out there that issues with heart disease and KD present later on. In fact, they can present very early in the diagnosis of KD. With that, I think that we'll close up the morning session. We're certainly available. We want to thank all of you that have been here live as well as virtually. And we look forward to seeing you all back next year. God willing, we will be here. Check out the KD Foundation website for our upcoming. And we thank you all for your attention today.